Sorry.
Okay. Hello. There is a that hammer on your left. That's for you. Yes. Yeah. All right. Good afternoon, friends. This afternoon we are going to start on transmission. The chair will be Dr. Lydia Mosi from the University of Ghana in Accra, Ghana. And she has been working on transmission work for Buruliosa for many years. I'm sure you will admire her chairpersonship. But I'm looking at all the skin NTDs and some of the issues around transmission. I think apart from Buruliosa and mycetoma, maybe the others, we have better understanding of how they are transmitted. Leprosy, I've put some question mark, but I think we know a bit. Tungiasis, my understanding is that we know how people get it. But the main challenging has been Buruliosa. So I think most of the presentations we will hear this afternoon will be around efforts to try to understand or better understand transmission of Buruliosa. I put this topic for this afternoon, hoping that there will be other diseases that have challenges and there will be submissions of other abstracts or topics for this, but it seems from the abstracts we received that transmission was not an issue for many of the other diseases, which means that their mode of transmission are well known. So let us hear what we have for this afternoon, and I hope that in a question that was put to Dr. Fahar this morning about how to prevent mycetoma, I think mycetoma too may be in the same transmission limbo like Buruliosa. There's some environmental human factor where the organism lives perhaps in the environment, but how it gets to people, I think, may also be a bit challenging. So let us learn from each other. And I give the floor now to Dr. Lydia Mosi.
Thank you, Kingsley, and good afternoon to everyone here. I hope you are well refreshed, and I'm looking forward to an exciting afternoon on transmission research. I also want to say a welcome to our online participants who are joining us via Zoom. Good afternoon to all of you. I'm very happy to be chairing this session, and thank you, Kingsley, for the opportunity. We have 8 talks in this session, with 1 keynote presentation that will be given by Dr. Tim Stineer. He will be telling us about understanding transmission of Buruliosa in Australia, progresses and challenges. I'll be really strict with my time enforcement because I've been given a gavel. I feel quite powerful, so please try to stick to your time. And Tim?
Thank you, Lydia. All right, we'll quickly move past that photo. Success. All right, well, Lydia, you are powerful even without your gavel. But so thank you. It is terrific to be able to present this work today on behalf of a large team in near southeast of Australia where I live. So this is understanding how Buruliosa is spread from the environment to people. And Professor Friedman on Monday introduced you to the astounding epidemiology of Buruliosa in southeast Australia. And this is just a cartoon of the epidemic curve. 342 cases confirmed in 2022. And on average, we are seeing about 300 cases a year of Buruli ulcer in the southeast of Australia. Around the major capital city of Melbourne, population of the state of Victoria is 5 million people. And with a growing focus in the nearby urban centre of Geelong. And these red spots here will remind you of the focal nature of the epidemiology of Buruli ulcer in our area, which is reminiscent of what we see in African countries. And one message, apart from describing to you how Buruli ulcer is being spread in our area, is that the bacterium causing the disease in Australia is not very different to the disease causing Buruli ulcer in African countries. And I think while the lessons we've learned in our area are not directly applicable in all situations, there will be major clues for understanding how Buruli ulcer is being spread in African endemic countries. Okay, let's move. So just some scene setting. We know from some very careful epidemiology led by the lead authors on these papers, of which Arvind Yaramilli, the last named paper there, is in the audience. We know that transmission peaks during the warmer summer months and that the incubation period is very long, 4 to 5 months. And this makes tracking epidemiological investigations very difficult, of course. But it also gives us a clue as to when disease transmission risk is highest, when the temperature is warmest. And the picture on the left, the human body there, is the careful mapping work led by Professor Paul Johnson and conducted by his talented medical student at the time, Arvind Yaramilli, showing the non-random distribution of ulcers on the human body. And it's these sort of lines of evidence, activity of the disease transmission in warmer months, this non-random pattern of ulcers on the human body, led Paul to the suggestion that perhaps insects, in particular mosquitoes, were playing a role in the spread of this disease. And so I am going to give you the punchline of my message here today. I'm going to tell you how Buruli ulcer is spread in southeast Australia. Then you can go back to emails and other essential duties. The 4 things you need to know is that Buruli ulcer needs some essential elements in order for us to see the disease in people. And those essential elements are a wildlife reservoir, the Australian native possum, which is exquisitely sensitive to Mycobacterium ulcerans infection. You need, the possum's infected with the bacteria, so that's the second thing. You need mosquitoes at a sufficient density in and around these infected possums, and you need people in the area where the possums and the mosquitoes are. And when these 4 factors coalesce, and perhaps there are some other risk factors, we see Buruli ulcer in humans. Now, I have one slide on possums, and there's a literature out there on the role of possums as a wildlife reservoir for Mycobacterium ulcerans in Australia. And I'm just going to summarise it in one slide, saying that the relationship between possums with Buruli ulcer, or shedding Mycobacterium ulcerans in their faecal pellets, the faecal pellets are shown in that image, the relationship between possums that have Mycobacterium ulcerans and humans with Buruli ulcer is so strong that we can build statistical models based on collecting possum excreta, carefully collecting it from around the endemic areas, building maps of where the excreta is positive, and then predicting a year in advance where humans are likely to get Buruli ulcer. So we have 2 things there, a powerful public health tool based on the very convenient analyte that is possum excreta, but also demonstration of that tight link between the wildlife reservoir and humans with Buruli ulcer. We have a submission under review at the moment that's describing this work, and there's a bioarchive preprint for those that are interested. Ooh, that is not good. Sorry, Stephanie, I apologise. Play? Something like that, yeah. Thank you. All right, 4 essential elements. Possums are a key thing. And next slide. Hang on, let's try. There we go. So I want to spend a few slides talking about mosquitoes because these are the vectors that are spreading the bacteria from the environment, from possums to humans. How do we know that mosquitoes are playing this role? I'm going to walk you through the evidence in a relatively compact amount of time, but again, there is some literature out there to support what I'm about to say, and we have some papers about to be submitted. Paul Johnson and colleagues noticed in their series of patients, and I acknowledge Paul Johnson for the provision of these images here that have been reproduced, the patient one in particular with permission, that the appearance of lesions on these parts of the body led Paul to consider that perhaps insects are playing a role here, in addition to the non-random distribution of lesions on the human body. And you can see here a mosquito landed on Paul's thumb in about roughly the position where one of his patients subsequently developed an ulcer. Anyway, this is all very interesting and curious observations, but how do we go to test the hypothesis that mosquitoes are spreading Buruli ulcer? In 2010, our colleague and renowned aquatic entomologist, Professor Rich Merritt, published a review with some others that are here at this meeting, explaining how if you want to implicate a vector, an insect in vectoring a disease, then you need to address some formal criteria. And there are several ways to do this, but essentially you are building a hierarchy of evidence to implicate your particular insect in the spread of a disease. And we followed, using Professor Merritt's recommendations, the Barnett criteria, and I've just summarised them there in these 4 criteria. The vector has close association with infected hosts. The vector, in this case the mosquito, must be found infected with the pathogen. The vector must be shown to acquire the pathogen from an identified source. And you must take your proposed vector and show in a controlled laboratory model that you can have efficient transmission to a susceptible host. So I'm going to quickly walk you through what we've done to address these criteria. And the first one, if I just go back one step, the vector has close association with infected hosts. We know that there are mosquitoes in very close association with the possums, which are highly adapted to life in urban Australia, and also the humans, us, that live in those areas. So I think criterion 1 is relatively easy to address. Criterion 2, we must find the vector infected with the pathogen. So to do this, we do surveys, environmental surveys. One large one was done in 2007, and the citation is at the bottom there, but we've repeated it, an even larger survey. 75,000 mosquitoes collected across the area shown in the map there on the right on the Mornington Peninsula. The take-home message from this investigation is that 7 per 1,000 mosquitoes we trapped were positive for Mycobacterium ulcerans. Now, if we compare that to what happened from Paul's survey in 2007, it's the same order of magnitude of positivity in the insects in a completely, well, not completely different, but in a different endemic area in the region. So repeated 10 years later, we find the same association with mosquitoes. We can go further, though, than what was done in those earlier studies. We can look in the insects to see what they have been feeding on. We can take the abdomen of mosquitoes that have blood-fed, and we can use genomics to sequence their contents and establish, using the methods of forensics, what those mosquitoes have blood-fed on. And the message from this slide is that we have trapped individual insects that have DNA from humans and DNA from possums in their gut. That is, an individual insect has fed on a possum and then fed on a human, or the other way around. This links possums and humans in the transmission chain with mosquitoes. The next piece of evidence, the vector must be shown to acquire the pathogen from an identified source. This is hard to do, but thanks to genomics, we have a technological approach to address this criterion. And what we've been able to do is, using very sensitive genomic methods, is sequence the pathogen genome, the genome of Mycobacterium ulcerans, from insects, mosquitoes, that are positive by PCR, and from possums. And this phylogeny laid across the endemic area is showing you that we have forensic fingerprinting genome matches between humans, possums, and mosquitoes from the same endemic area. I'm nervous to click this again, Stephanie. We can take it even further. We can look at the spatial clustering of these three elements. We could look at where there are non-random clusters of human cases of Buruli ulcer, non-random clusters of possums that are shedding Mycobacterium ulcerans, non-random clusters of mosquitoes that are positive for Mycobacterium ulcerans, and we find that they overlap. And this is spatial scanning statistical analysis, and that inset box there showing with those three overlapping circles in the three different colours is where each of those elements overlap and intersect. So again, showing that there's a close relationship between these three elements. And finally, I don't have time to dwell on this today, but in previous work led by Professor John Wallace at Millersville University in the US, we've done some first experiments to demonstrate using experimental mice and insects that we can, in the laboratory, show how transmission may occur with an infected mosquito. Next, oh, no, I don't, sorry, Stephanie, I can probably do it. Oh, back one, sorry. Yeah. So for my colleague on Monday who raised the idea that we should be thinking about some of these neglected tropical skin diseases in the context of One Health, well, I couldn't agree more. What you need to, I think, leave this room knowing is that Buruli ulcer is a zoonosis, at least in Southeast Australia, and I would put some money on it being a zoonosis in other endemic countries. So it's a zoonosis, it has a major wildlife reservoir, and as you'll hear perhaps today, there may be other wildlife reservoirs, but possum is the major wildlife reservoir. Mosquito is the primary vector for spreading Mycobacterium ulcerans to humans. We have work to do to understand the exact way of the link between possums, mosquitoes and humans, but that's work to be done. And we have a project trying to address this. This talk included progress and challenges. I see that we have one major challenge. I would like to show that if we if we undertook a control, in particular a mosquito intervention, that we could impact the high number of human cases that we're seeing, thereby proving that insects, mosquitoes are spreading Mycobacterium ulcerans to humans and demonstrating a very powerful public health intervention. Although I would just flag that today you'll hear some interesting ideas on alternative interventions based on this transmission model that I've presented. Next slide, please. Thanks, Stephanie. A large body of people to thank. Paul Johnson has led much of this research for the last almost 30 years, and oh, we've lost the acknowledgement slide. Is it possible to get it back, Stephanie? La diapo de tous les gens qui font le travail. That last slide, no? Can you go back? Oh. Look, just while a large team involved in this over decades now, and I would just like to highlight a few. Paul Johnson, Koen van der Lenutte, who's done a lot of the possum excreta survey work, Andrew Bulgens, who is at this meeting online. I also would like to particularly acknowledge Elaine Tay from the Department of Health in Victoria, who looks after Mycobacterium ulcerans epidemiology. Without the epidemiology, we would really be lost. And I'd also like to acknowledge two people who are not on this version of my PowerPoint presentation, Dr. Emma Hobbs, who has done much of our possum work, and Professor Anna Meredith, who's supervised Emma. So thank you, and I will leave it there.
Thank you, Tim, on this presentation and showing us through the years how you've tried to build on this Barnett model to provide salient evidence to help us understand transmission of BU and the involvement of mosquitoes. I'm sure to bring up some interesting discussions at the end on whether it's mechanical or biological, so I'm looking forward to hearing some of these. Next, I'd like to invite Delini Wijiskara, who is presenting online via Zoom. She's presenting on on the title, Transmission Status of Neglected Tropical Diseases with Skin Manifestations in Sri Lanka. So, Jalini, if you're on.
Chairperson, ladies and gentlemen, it's a pleasure to be with you today. I'll be presenting transmission status of neglected tropical diseases with skin manifestations in Sri Lanka. I am Dr. Devini Vijayasekara, consultant community physician from the Anti-Leprosy Campaign Sri Lanka. Other two co-authors are Dr. Indivari and Dr. Pubudu, are from the Anti-Filariasis Campaign and Anti-Malaria Campaign. Next slide, please. This is our outline of our presentation. It will include introduction, objective, methods, results, main challenges we are facing, conclusion, key take-home messages, and of course, last acknowledgement. So, 3 neglected tropical diseases in Sri Lanka are associated with skin manifestation: lymphatic filariasis, leprosy, and leishmaniasis. Out of these, lymphatic filariasis has been eliminated as a public health problem in 2016, as the microfilariasia rate was less than 1% for decades, and leprosy has been eliminated as a public health problem in 1995, as the case on treatment was less than 1 per 10,000 population. Since elimination of both diseases show a low-grade transmission, and now both control programs have embarked on total transmission interruption. Trend predictions show that total transmission interruption would be possible by 2023 for filariasis, and of course for leprosy 2027. Even though the zero transmission would be achieved within few years, program targets have been set giving allowance for the low screening rates with current crisis situation within the country. The disease leishmaniasis is still on the control phase, and it's a newly re-emerging disease in Sri Lanka, and more attention to be paid for the control of this illness. In objectives, the main objectives of the lymphatic filariasis and leprosy program are to interrupt transmission of the disease by 2030. Objectives of the leishmaniasis control program is to reduce the incidence of the disease from 7.2 per 100,000 population to less than 1 per 100,000 population by 2030. Methods of achieving these targets are different in different disease programs. In Sri Lankan system, lymphatic filariasis and leprosy has designated disease control programs, namely Anti-Filariasis Campaign and Anti-Leprosy Campaign. These campaigns have dedicated staff, including grassroots level workers for disease prevention, control, and surveillance. This is the strength for reaching elimination targets in Sri Lanka. Other than this, lymphatic filariasis program follows mass drug administration, which help further reduction of microfilariasia rate to a very low level. Sri Lanka used 2 drug regime along with DEC and albendazole for mass drug administration. Further, anti-filariasis campaign has established morbidity management clinics in all endemic districts to manage patients with chronic complications and skin manifestation. Leprosy elimination was achieved by alleviating stigma and improving awareness through an effective social marketing campaign. This led to more case reduction. After achieving elimination, leprosy program was integrated into the general curative healthcare service in Sri Lanka, and now the patients are followed up by the skin clinics in curative sector. Leishmaniasis does not have a separate control program at present. Patients are identified with passive presentation to curative sector. Recently, anti-malaria campaign in Sri Lanka taken up the vector control and control component of leishmaniasis. For all these 3 diseases, Ministry of Health is maintaining disease vector and parasitological surveillance databases. They are used to assess the burden distribution and monitor the trends. In this abstract, we will be presenting results of secondary data analysis of data in published report. Results will be presenting numbers, percentages, and graph. Results in lymphatic filariasis. Lymphatic filariasis is endemic in 8 districts in 3 provinces: Western, Northern Western, and Southern and Southeastern coastal belt of Sri Lanka. Sri Lanka maintained microfilaria rate below 0.05 since stopping mass drug administration in 2006. Continuous low-grade transmission is observed during post-validation surveillance period. Mosquito surveillance is a sensitive indicator for transmission. Sri Lankan mosquito surveillance data shows high mosquitoes product positivity, which says still we have a higher transmission potential. Reporting of child cases, children less than 18 years make 21% of the total caseload. These majority of them are males. Other than these indicators, the morbidity management clinic receive more patients with chronic complication of filariasis. On average, 600 to 700 patients referred to our clinic. And all these data shows continuous transmission of filariasis even after validation as a country eliminated filariasis as a public health problem. So in leprosy, more than half of the leprosy cases report from Western and Eastern province. A decline trend is observed in new case detection, but percentage of child leprosy cases and the percentage of grade 2 disabilities were being static for the past 2 decades. The child leprosy case percentage fluctuate around 10, and of course, the grade 2 disability fluctuate around 5 to 7%. Higher percentage of child case is a proxy indicator of high active transmission of the illness in the country. Majority of the reporting patients are economically active male patients. Next slide, please. So in leishmaniasis, for the period of 2001 to 2019, a total of 19,361 confirmed leishmaniasis patients were identified from country. There is an increased trend of detecting the patients. 6 districts report more than 1,000 cases, mainly from central, north central provinces. The vector indicator research shows abundant sandflies, which increase the transmission of the illness. Next slide, please. So in conclusion and recommendation, low growth transmission occurs in leprosy and lymphatic filariasis for decades after elimination in Sri Lanka. Elimination as a public health problem do not warrant natural dying of the disease. More intensive interventions together with integration with health and non-health organizations are required to reach total transmission interruption. Next slide, please. So our main challenges. Night blood filming is the method used for screening filariasis, and it is having a low sensitivity. Therefore, missing of true cases is a problem, and risk introduction of cases via the migrant workers is another challenge at present. Low community compliance for the screening, thinking that filariasis is an eliminated disease is another challenge at present. Of course, for leprosy is still a stigmatized disease in the country. It's a major challenge for elimination of the illness. The lack of awareness among the public is also causing problem in identification, treating at present. Leishmaniasis is a newly emerged disease in the country. Therefore, lack of effective control methods and underdevelopment of the referral system to refer patients to the dermatology clinics are main challenges at present. Apart from these, the current financial crisis is a big challenge for the preventive activities at present in Sri Lanka. So the key take-home messages. Next slide, please. Finally, the key take-home messages: elimination of a neglected tropical disease as a public health problem does not warrant total interruption or transmission of the illness. Therefore, the surveillance needs to be strengthened to identify the cases, especially during the post-validation period. We all know that last lap is the most difficult one. Hence, more investment is needed during this phase. The present strategy needed to be revisited, and identification and introduction of novel strategies is a must during the post-elimination period. And more research is needed to identify effective methods to control the sand flies in the country. So at last, the acknowledgement. We would like to acknowledge the Ministry of Health Sri Lanka for sustaining financial allocation for a disease of leprosy and leishmaniasis that have been eliminated as a public health problem in the country. Thank you.
Thank you, Delini, for that wonderful presentation and for really highlighting the need for active case surveillance in our transmission efforts. And thank you again for being ahead of time. It gives us more room to have discussions later on. Next, I'd like to invite Stephen Muhi to give a presentation on establishing a controlled human infection model of Buruli ulcer. So Stephen, from the University of Melbourne, the floor is yours.
Thank you, Lydia. So nice to meet everyone. My name is Steve. I am an infectious diseases specialist at the Royal Melbourne Hospital and a PhD candidate under Tim Stenier. So I'd like to introduce my work. It's not a standard presentation in terms of the research that I've been doing, but it's very hard to succinctly put a year and a half of research into 15 slides, but I'll do my best and hopefully the technology will keep up with us. So the title of my talk is Establishing a Controlled Human Infection Model of Buruli Ulcer. The overarching aim of this presentation is to introduce the concept of a deliberate human infection with Mycobacterium ulcerans in order to study multiple aspects of the disease. I've got a few other aims that I'll talk about later, but hopefully you'll understand a brief summary of why, the rationale, why we're trying to do this. So in terms of the background, what is a controlled human infection model? As the name suggests, it's the deliberate infection of human volunteers in a controlled, careful fashion to reproduce some typical disease phenotype. These models are very interesting and they're more relevant for complex but uncommon pathogens such as Mycobacterium ulcerans, and generally they can only be established for non-severe disease syndromes. They're self-limited or there's some sort of reliable treatment option and rare rates of complications. Human models have been safely established for a number of different pathogens already that have the possibility of causing severe disease syndromes, including falciparum malaria, COVID-19, cholera, strep pyogenes. But compared to these pathogens, Buruli ulcers are typically slower, less severe, relatively non-invasive, associated with lower mortality rates, and usually restricted to skin and subcutaneous tissue. But nevertheless, safety is absolutely paramount with these studies, which stresses a cautious approach to strain selection and manufacture, the careful selection of consenting adult volunteers according to some defined eligibility criteria that has been approved by a Human Ethics Committee. It requires close clinical monitoring in order to support patients throughout the study, with prompt administration of curative antibiotics at the study endpoint and oversight by an independent safety committee. So what is the use case for an M. ulcerans controlled human infection model? Well, it's useful to test a number of potential prophylactic or therapeutic interventions, particularly vaccines, because there's really no clear path towards testing efficacy of vaccines. This is mostly due to the sporadic and highly focal nature of Buruli ulcer. In terms of its epidemiology, as Tim and Deb have already discussed, and therefore clinical trials are likely to be underpowered and very expensive, and in fact will probably expose thousands of people to an experimental intervention with no good result. So a Buruli ulcer CHIM, or controlled human infection model, could potentially provide a biological safety of biological signal of vaccine safety, identify immunological correlates of protection or disease, and then stratify subsequent vaccine development. So, for example, to test a vaccine with 70% efficacy, if the CHIM had a 100% attack rate, it would require just 14 participants, so 7 participants per arm. But in summary, the model has the potential to revolutionise Buruli ulcer research. My question for the last 18 months has really been, what does the ideal challenge strain look like? I think it looks like that, but we have published, or we've submitted a paper for peer review at the moment, which will go into a bit more detail. Hopefully you'll be able to read that in the coming weeks or months. So I'm going to spend a bit of time talking about the characteristics of an ideal challenge strain. You wouldn't necessarily want to be a participant getting inoculated with a strain that was associated with certain severe clinical disease syndromes. So that's the first thing to start off with. It shouldn't be associated with severe disease. It should reproduce a typical infection phenotype so we can actually understand it in a standard model. We should be able to biologically have some plausible way of inoculating our participants. It should be susceptible to antibiotics before we have to treat the participant. We should be able to culture it in something that's not toxic to people. It's animal-free because we want to reduce the risk of bovine spongiform encephalopathies or mad cow disease, because most of the media that we use to culture tuberculosis and other mycobacterial organisms in the lab contain albumin and other animal-derived products. Ideally, it should be free from genetic modification or chemical modification, so it's a standard model. We should be able to be fairly certain as to the dose we're giving our participants. We should make sure that after we freeze it, we thaw it, it's still viable. It needs to produce the key virulence factor, which is mycolactone. It needs to actually have the genes encoding antigens of interest for vaccines so that we can test those vaccines, and it has to be genetically stable so it doesn't mutate into some severe, some variant. And also we need to actually prove that we can look at all of those different criteria and test it in a real world scenario. So to start off with, that first category is not associated with clinical disease. Well, Australian and African isolates belong to a classical lineage together, so it provides a useful comparison. In Point Lonsdale, which Deb mentioned the other day, but it's one of those parts in southeastern Australia, in Victoria, there was an outbreak in 2002 with 79 infections that were reported. And a larger study of 180 cases across the entire region, which includes Point Lonsdale, showed that the vast majority of people in that region had a single lesion, and most of them were just a nodule or an ulcer. A very small proportion of them presented with an edematous or an acute inflamed phenotype, and most of them were of advanced age. Only one person in that particular retrospective study had septic bursitis or inflammation of the bursa in the joint, and one had osteomyelitis or infection of the bone. And that contrasts with rates of as high as 6% in Benin due to multiple different factors. But interestingly enough, a paper published in the past showed that osteomyelitis in patients in Benin could actually be very distant from lesions, superficial skin Buruli ulcer lesions. And in about 1/4 of people with osteomyelitis in Benin, none of them actually had a history of Buruli ulcers. And that may actually represent hematogenic seeding, which is very rarely reported in Australian isolates. And it could be that African isolates are a little bit more tolerant to temperatures that we don't see in Australia, for example. So firstly, that's a pretty important aspect of not being epidemiologically associated with severe disease. This is that isolate I showed a picture of at the start, JKD 8049. It was isolated during that outbreak in Point Lonsdale in Victoria, and this was isolated from a human patient. It's a clinical sample, and that person presented with a typical, slowly progressive, painless ulcer at the back of their calf. So in its own previous history, it wasn't associated with severe disease. Secondly, it needs to reproduce a typical phenotype of disease, and this is Arvind Tiramelli's picture, which we've seen 3 or 4 times now. But firstly, it needs to be generalisable, and in Australia, the most common site is between the knee and the ankle, and this is a reasonably good challenge site because it avoids the joints, so it reduces the risk of contractures. It may be more amenable to surgical excision, and it result in less obvious scarring to the person because it's towards the back of their body. And it's less likely to be associated with an edematous phenotype as well, which we want to avoid. Realistic doses of the bacteria are actually required to inoculate mice in a particular study that was done in the past by colleagues here. Most studies in inoculating mice have actually been done with extremely high doses. So when I say CFU there, that's colony forming units, but I guess that replicates, or the idea is that a CFU represents a single bacteria. So previous studies have actually used very high doses of 10,000 to 1 million doses of bacteria when they've done challenge studies looking at vaccine efficacy. But in fact, for this particular isolate, JKD 8049, The dose required to infect 50% of mice in a particular study was just 2.6 bacteria. So we have probably been completely overwhelming the host immunity and underestimating the vaccine efficacy in previous studies. And the study endpoint should be realistic and generalisable. So we suggest that at the particular moment of onset of ulceration, or if there's some a pre-ulcerative nodule or small lesion, then that would be the end point of the particular study. So it wouldn't be allowed to progress to severe disease or Hood stage 2 or 3 disease. And obviously, if it presented with a more acute subtype, it would require rescue antibiotic therapy. Thirdly, it needs to be amenable to some biologically plausible route of entry. And this particular strain of M. ulcerans has been shown to be successful in establishing infection in mice, as well as in two different ways, using both a mosquito bite model as well as a subcutaneous infection model. And so it does provide some biologically valid model of infection. And using a needle stick puncture in two different studies, they've both shown that very low doses of Mycobacterium ulcerans have been successful in producing infection in these particular studies. So the proposal is therefore to inject a very low number of bacteria, approximately 30 bacteria, subcutaneously into a human participant, but minor deviations away from this that are a little bit higher than this are probably not clinically significant. It needs to be susceptible to clinically relevant antibiotics, and I have shown in this study using a particular modality called mycobacterial growth indicator tubes, which still need to be validated by a second external laboratory. But I have shown that this particular isolate is susceptible to rifampicin, clarithromycin, and ciprofloxacin in an in vitro model, and looking at other isolates as well. So I think that's pretty useful. It needs to be culturable in a non-toxic medium, and this is a particular medium that I created called Sorton's medium with vegetones, so something to supplement it to enrich its growth. On the left, you can see what it looks like in the lab, so welcome to Tim Stenier's lab. This is where I've inoculated it into this medium, and then a few months later, I've got a huge amount of growth. The problem with this particular organism, it's very slow, it's very clumpy, it's a real pain in the neck to try to manipulate. These clumps are very difficult to break up. They stick to everything and they get stuck in needles. But I can show that not only is M. ulcerans easy to grow, but it's easy to grow on a solid medium along with other different mycobacterial pathogens. This is one of those clumps that's seen under a Ziehl-Neelsen stain, and you can see there's thousands of bacteria there. If I inoculate one person accidentally with one of these clumps, I may be severely overdosing them. So I need to be able to make sure I can accurately declump these particular suspensions. So I've come up with an innovative strategy where I've put them in this shaking model for 3 months. They shake for 3 months. So you can understand what a patient scientist I have to have become. 3 replicates of the same bacterial suspension, and it's very hard to grow them in a turbid fashion, but I was successfully able to do that. Then to get rid of all of the clumps, I've actually filtered it through this 5 micrometre pore filter to absolutely remove any clumps from that particular suspension. And you can see on the left, I've put some of those spots onto an agar plate, and you can see it grows irregularly and clumpy. But on the right, you can see it grows after I filtered it in a nice homogeneous suspension. So I've removed all of those clumps. The dose is very important. So in the middle, you can see some collaborators infected a mouse's ear with about 80 bacteria, and on the right with 10 times the dose, and it results in more rapid necrosis and tissue breakdown and tissue loss. So looking at it under a microscope, I can be 100% certain that I've removed all of the clumps to avoid the possibility of overdosing my participants. On the left, you can see a large clump, and on the right, I've looked through 50 high-power fields, and I'm absolutely certain that I don't have any clumps visible there. So I'm reassured by the safety of that process. And not only that, but I can say that I can grow it after I've declumped it, and I've lost about 75%, 74% of my bacteria, but I've got a good amount afterwards. Then what I do is I freeze it, and then I thaw it. And I can show you that after thawing it, after freezing it and thawing it, I don't lose any mycobacteria in the process, and there's no statistical significance in this process between those different time points. So I'm successfully able to do that as well. To wrap up, I've shown that it does produce mycolactone, that key virulence factor, when I grow it in this particular media. It does have those genes that I need it to encode for those different antigens, and it's yet to be shown that it's genetically stable. But the next steps are that I need to demonstrate in a human macrophage model that I can successfully stimulate an immune response. We're moving on to a possum challenge model, which Dan will talk about shortly in today's session. And finally, we'll talk another time about a mouse tail model, which hopefully will be able to prove that my particular suspension that's frozen and thawed is successful in actually stimulating or generating an infection. And this is some images of the macrophage line there with GFP. So there's a lot there to summarise in a year and a half of work, but hopefully I'll be able to stick around later in the afternoon to talk to others who might have some questions. But there's a huge range of different labs that were involved in this research so far, lots of people to thank, particularly Tim Stenier, who's let me do this work in his lab, the team at the Doherty Institute and Royal Melbourne Hospital, my co-supervisor Mark Pellegrini at WeHi, and Josh Osowicki at the MCRI. Thanks very much.
Thank you, Stephen, for that very insightful presentation. It's definitely one of those things that we need to talk about more on getting that one strain that every single lab can rely on and use so that we can reproduce each other's work. It's also very interesting for us to open up discussions later on whether the infection goes through the routine BU hallmark stages of nodule, edema, plaque, and then eventually an ulcer. So thank you so much for that work.
Thanks, Lydia.
So next, I'd like to invite Paul Johnson from Australia as well to give us a talk on peak transmission of Ross River, Barmah Forest virus infections, and Buruli ulcer in temperate Victoria are aligned and strongly influenced by season. Paul, the floor is yours.
Thank you very much for the opportunity to speak, and good afternoon to everyone. So Tim's fortunately covered this already, but I just want to draw your attention to the fact that the data I am presenting here relates to the temperate part of Australia. We come from Victoria, which is right down the bottom, that little triangular southeastern part of the country, where we have this steepling increase in Buruli ulcer year to year, almost exponential, that curve, and that's all in Victoria. Now, we have a typical temperate climate so that our winters are cold and we can even go skiing. And that's important to remember because the temperature has a big effect on both human and insect behaviour. Also, you've probably seen this figure before on the other side of the slide, but it shows the endemic peninsula, the Mornington Peninsula, the Bellarine Peninsula, and then the big city of Melbourne and of Geelong, where we are now seeing outbreaks within our suburbs as well as along those two peninsulas. Now, Tim's already covered this, but we've done these extensive possum excreta surveys, which show that contamination of the environment is frequent, not evenly distributed, and that we're now able to use possum excreta surveys to predict human cases and show a very strong association between PCR positive excreta on the ground and the presence of Buruli ulcer in humans. The possums themselves get the disease as well as excreting it. I just wanted to say about that slide that I don't have time to cover the data, but the possum excreta signal persists throughout the year. So while it's uneven, it is detectable all through the year in the winter, summer, autumn and spring. So to the study, we have got this rapid increase and we have yet been unable to really organise a specific targeted campaign of prevention because it's not been agreed what the main mode of transmission is. We do have increasing evidence that mosquitoes are involved, but is this just one of several ways to get BU? Because if you put all your eggs in the mosquito basket and it only explains 10% of cases, then you haven't really achieved anything from a public health perspective. Now, Ross River virus and Barmah Forest virus are alpha viruses that are agreed and known to be mosquito vectored, and they also cause outbreaks of disease in Victoria we see seasonal polyarthritis with rash, and they can be quite debilitating, but I won't go into the details about this disease. The main point is that that's also it is acquired from mosquitoes, and it is also seasonal. So the objectives of the study was to investigate whether there is a temporal association between the incidence of borreliosa and mosquito-vectored alphavirus infections, which is Ross River virus and Barmah forest virus in Victoria. And the methods were relatively simple. We used the Victorian disease notification data by month, which was accessed through the Victorian Department of Health for the whole state. We looked at month of notification for borreliosa and the two alpha viruses combined, that's Ross River virus and Barmah forest virus, over a 6-year period from 2017 to 2022. And I need to say that we're fortunate in Victoria and Australia to have universal health system. We have easy access to doctors and pathology. And for our notifiable conditions, they're typically now notified electronically. So there's very little delay between a positive test and the Department of Health receiving a notification. What we can't control for here is the time between when a person finds there's something wrong with them and going to a doctor and getting the diagnostic test. We've just assumed this is a random variable, which we don't account for, but we suspect that it's probably shorter for the alpha viruses than it is for borreliosa because they are more dramatic with fever, rash, and joint pain. Now, here's the first results slide. I'll just take you through it carefully so you can see on the bottom axis the years, the 6 years of the study, 2017, 18, 19, 20 to 22. You can see that each column represents 1 month of each of those years. It's also divided by seasons so that we can see summer, autumn to the left of each year and winter, spring to the right. So those are the months of December to May and January to November. And just reminding you once again, we're in the southern hemisphere. The winter solstice occurs in June and our coldest months are July and August. Now, if we just look, and the other thing to note is that the axis varies between the two conditions. So it's 10-fold higher. That's the vertical axis for the alpha virus infections at the top compared with Borrelia at the bottom. Now, if we just look at the alpha viruses, you can see this very significant year-to-year variation. Some years we have hardly any, and other years we have big outbreaks. So it's difficult to sort of describe a pattern there. But one thing that we can agree on visually is that all the notifications occur in summer and autumn, and there's almost none in winter and spring. However, if we go down and look at the borreliosa in black, you can see there's much less year-to-year variation. There's certainly very significant variation during the year by season, but it's actually quite different from the alpha viruses so that peak notification occurs in the winter and spring instead of in the summer and autumn. So I think we can safely conclude without doing a statistical analysis, there's absolutely no relationship and we should now go home. However, we need to consider the incubation period. Now we know pretty clearly that the incubation period of Borrelia in Victoria is 4.5 to 5 months. That's the median and the mean. It's normally distributed. But that the incubation periods for the alpha viruses are much shorter, typically 7 to 14 days. This allows you to infer peak transmission by subtracting the incubation period from the notification time to get an idea of when peak transmission's occurring. Now, in this graph, I put both the two conditions on the same axis, which is interrupted because of the much higher numbers of alpha virus infections. And I think we could agree visually that now there is a significant alignment where the two conditions appear to be transmitted at the same time. Now, it's been quite difficult to statistically analyse this, and so far it's defeated me, but I'm not a statistician. But we can have a look at it just qualitatively. So what I've done here is blown up the bottom of the axis, and you can see that we would all agree that the transmission is consistently lowest in winter and spring for both the alpha viruses, which we know are vectored by mosquitoes and Buruli, and the converse applies, that they peak in the summer and autumn. So our conclusions. Others have shown that M. ulcerans contaminates endemic areas throughout the year. I shouldn't say others because I'm part of that team. We have shown that. So the presence in the environment per se could not explain that very marked seasonal variation that we see. We have extensive evidence that mosquitoes trapped in Buruli endemic areas carry M. ulcerans, as Tim has shown, and there's plenty else to go with that. We know that alpha viruses are transmitted by mosquitoes, but not throughout the year. And I've shown you that transmission maxima and minima for Buruli ulcer and alpha viruses correlate. We think that mosquito abundance is the major driver of Buruli transmission in Victoria. And we think that a public health campaign focused on mosquito avoidance is likely to reduce the incidence of Buruli ulcer in Victoria. So my take-home message for Buruli in Victoria is to avoid mosquitoes. Thank you.
Thank you, Paul, for that presentation. And I mean, this talk raises a very, very important and critical subject of taking a look in retrospect back at all our transmission research under the lens of the incubation period, because it really would make a significant difference in some of the predictions that we've gone. Thank you for that. So the last talk for this first half of this afternoon is by Jean-Francois, and he will be talking to us on the evolutionary ecology literacy is important for improving the understanding and management of mycobacterial skin diseases. Jean-Francois?
Much. So this presentation, this presentation is roughly the same as the one presented by Christine Chevillon during the Buruli ulcer side meeting this morning. So people from the Buruli ulcer group can get out and have a nice cup of coffee or tea, and other people can stay, of course. So this is also a non-standard presentation because we don't have any new results to present you. I mean, the results are coming soon, will be published within the year or next year. But what we want to do with this presentation is to share with you the experience and viewpoints by disease ecologists and evolutionary biologists about what is a disease, being a plant, a human, or an animal disease, what is virulence, and what is pathogenicity. This is the main idea of this oral presentation today. So our objective was, through a bibliometric work, to examine how the evolutionary ecology literacy on infectious disease systems may improve the understanding and management of mycobacterial diseases. So in general, in medical and veterinary perspectives and research, studies on mycobacterial disease just focus on vertebrate infections only, on discriminating between two groups of mycobacterial pathogens. On the one hand, the Mycobacterium tuberculosis complex, so the so-called MTC or MTBC clade, characterized by host-to-host transmission and the fact to be an obligate specific pathogens. So this is illustrated on the left side of this slide. So on the right side, and on the other hand, opportunistic pathogens with saprophytic water, plant root, or soil life stages being the infectious stages. The absence of saprophytic life stage and the fact that it was long only reported in humans classifies Mycobacterium leprae with the tuberculous MTBC group. The evolution of infectious disease agents towards complete virulence while adapting to new hosts could be expected, but this is a very rare event that is observed in disease ecology. In many cases, evolutionary biologists have showed that the maintenance of virulence is maintained through time because of relying on the same mechanisms than host-to-host pathogen transmission, so that any optimization of host-to-host transmission indirectly selects for virulence, and this is what we call the transmission virulence hypothesis that we will here refer to as being adaptive virulence. On the other hand, the coincidental evolution hypothesis refers to cases where virulence arose as a coincidental byproduct of the adaptation conferred by the virulence factors to other components of the ecological niche of the pathogen, so being, for instance, the avoidance of predation by protists or the adaptation to some environmental host reservoir. So, I mean, the circumstances, the environmental circumstances, and the different, I mean, guys surviving in the same system as the mycobacteria are very important indeed to understand for understanding virulence in mycobacteria. So how do these concepts connect with the diversity in mycobacterial pathogens? The questions of the source of selection for mycobacterial pathogenicity and the sustainability of their infectious source through time and space. Most non-tuberculous mycobacterial infections of vertebrates do not result in transmission of the pathogen from the infected vertebrate to the environment, making thus the coincidental virulence hypothesis as the most likely origin of their pathogenicity. And this is illustrated on the right side of this slide. As observed on other opportunistic bacterial pathogens, we also expect some links between the competition for resources and pathogenicity. The possible existence of environmental host reservoirs open the possibility that some of these hosts are driving the pathogenicity of the non-tuberculous mycobacterial infections through space and time. Adaptive virulence is a verified scenario on the left side of this slide for the Mycobacterium tuberculosis complex or clade. A remark worthy of note on tuberculous bacilli, clonal lineages may infect diverse vertebrates, but each is highly specialized for completing its life cycle, thus for promoting disease and achieving transmission. Direct host-to-host transmission and adaptive virulence leads thus to a co-diversification of the tuberculous clones with their hosts. The adaptive virulence scenario may also apply to other mycobacterial agents of pathogenicity as well. M. leprae. Mycobacterium leprae is not cultivable, characterized by massive gene decay and low off-cell survival. Spread among people is thought to be achieved through coughing and contact of nasal fluids with an infected symptomatic or pre-symptomatic person, but transmission requires extensive contacts with these infected people. However, there are reports of nasal carriage of infection in a wide diversity of animals. For instance, Lepra bubalorum was described as a possible reservoir of Mycobacterium leprae in Indonesian water buffalo. These two first points indicate really that the probable existence of some environmental reservoirs for that bacterium. Moreover, Mycobacterium leprae circulate among wild animals so that Eurasian red squirrels in the British Isles, armadillos in the Americas, as well as in remote wild chimpanzees population in Western Africa. Assuming the same evolution as MTBC group, we expect genetic differentiation among the strains of Mycobacterium leprae circulating in distinct hosts. This next analysis, if we want to come, this analysis shows a very different pattern than that observed for the tuberculosis complex. In this tree on the left, lineages are color-coded, and animal figures indicate that strains circulating in animals, in squirrels, for instance, those circulating in European red squirrels are direct descent of the bacilli that used to circulate in British human people in the Middle Ages, before the disappearance of human leprosy in Europe, probably driven by changes in human behavior. In the Americas, there is no evidence of host tropisms in Mycobacterium leprae, and genetic divergence between the bacilli infecting either human or armadillos. By contrast, the emerging picture is a clonal expansion of bacilli circulating across animals and humans, including thus recurrent and reciprocal host shift between at least 9 bounded armadillos and humans. Overall, the emerging pattern pleads for the coincidental virulence hypothesis for Mycobacterium leprae. Mycobacterium leprae likely acquired its particular genomic features and pathogenicity through adaptation to intracellular life in a non-vertebrate host that was to be discovered. The authors note in this paper that Mycobacterium leprae could be an endosymbiont of some protists or other types of organisms. They also note that arthropod-borne transmission of Mycobacterium leprae has never been fully ruled out as plausible alternative to direct human-to-human transmission. Phylogenetic surveys did not support either the usual assumption of host specialization in Mycobacterium avium paratuberculosis, which is an animal disease as well. So the same trend is observed for this Mycobacterium avium tuberculosis indeed. Mycobacterium ulcerans, or MU, which is of concern here, is the only species for which the question of the maintenance of infectious sources through time and space in the field have raised scientific interest. Numerous studies have evidence that MU can be associated to a wide array of species across the intertropical belt in search of a possible ultimate reservoirs. What's look interesting here to stress here is the information that was gathered from studies that have analyzed the circulation of Mycobacterium ulcerans among all the ecological niches MU can occupy within aquatic sites, including the soil, water, plant roots as well. These studies are thus based on systematic sampling on the understanding of what really exists, so we open the window to get a bigger picture of all possible organisms to which MU can be associated to in the field. Systematic screening for the presence or absence of MU among those samples, and replicated sampling among aquatic sites that either relying on human infection cases or not. These studies evidence that various factors are playing a key role in the maintenance of MU infections, and these factors are operating at various scales. This can be pH, this can be seasonal fluctuation, this can be regional factors like landscape, which is very important as well for the maintenance and spread of MU in aquatic environment, notably in Western and Central Africa. So I will skip these slides to go to the end of this talk. Laboratory studies confirmed the functional link between aquatic insects and MU abundance. Indeed, the addition of chitin in medium growth culture boosts the MU growth. Moreover, the addition of chitin has also shown to increase the pH tolerance range for MU. At very low pH, usually preventing MU growth, the addition of chitin did drastically stimulated bacterial growth. So, etc. So I need to skip some slides to go to the conclusion to respect my time. Now, research in disease ecology. Or maybe you are not familiar with publication published in ecological or biogeographical journal. I'm sorry. Yeah, go. Okay. Now, research in disease ecology analyzed the geographical distribution of soil-dwelling bacteria and fungi, as illustrated on this slide. These authors evidence that a cluster A, here, cluster A, I'm sorry, previous one, the previous one, well, cluster A, this is in the middle of this slide, cluster A of microbial species is very likely to expand their distribution with global change, and notably with climate warming and land use change, just because this type of mycobacteria are embedded into soil, fresh waters, and plant roots as well. Mycobacterium species are part of this cluster A, indicating that thus that new still unknown mycobacterial species will potentially invade new areas due to climate change and soil erosion notably, and thus establish new contacts with vertebrate species, being animals or humans as well. We can thus expect future outbreaks of mycobacterial disease in the future. And to conclude, and the take-home message are, we saw that adaptive processes have rarely played a role in vertebrate mycobacterial interactions, but that through coincidental adaptation, diverse mycobacteria are pathogenic. So the environment is strongly important for the genesis and the development of pathogenicity. That is to say, they are already armed to attack vertebrate species. Therefore, the understanding and management of mycobacterial diseases require analyzing the circulation of such opportunistic pathogens among non-vertebrate organisms that are totally embedded into natural ecosystems and that may provide indicators of possible new outbreaks or do play a role in the persistence and dispersion of the pathogens through time and space, but also in impacting its pathogenicity levels, indeed. And next slide. We finally thank the different foundations that funded this work and the many people who contributed and participate and helped to develop this work. So thank you very much for your attention. I'm sorry, I must just apologize, present you my apologize, because I have to leave to reach Montpellier for a series of meetings tomorrow and the day after, and notably with the World Organization on Animal Health located in Paris. Yeah. Thank you very much.
Thank you, Jean-Francois, for your presentation. And as he's requested, if you have any more questions or engagements with him, please contact him via email. His email address is in the participant list. But thank you for that presentation, which has raised really interesting questions on not only the evolution of emulsurants and other mycobacterial species, but the fact that introduction of the mycobacteria is important, its survival and its maintenance is also really important in both virulence and pathogenicity of the organisms. So at this point, we have to open the floor for discussions. I would like to invite all the presenters, if possible, to log into the Zoom because there are some questions on there which are quite specific for you. And I'll probably start with the room. So if there are any questions in the room, I would invite you. So in my peripheral, I think I saw your hand up first. So Dr. Dang?
Oh, yeah.
Yeah.
Thank you, Madam Chairperson. A very interesting session, really interesting session, highlighting the transmission dynamics of Brule ulcer. So I have a couple of questions. One, with regard to the vector, you kept in all of the presentation, I hear that only mosquitoes again and again. So I was wondering, are all species of mosquitoes potential vectors? Because I saw in one of your presentation, one species, you found positive in one species, but I was wondering whether specific species are incriminated or all species are capable. That's one. The second is possum is okay, repeatedly again as a reservoir mentioned, but how about other mammalian reservoirs? This is also another question that I would like. I don't know if there are similar studies with regard to this transmission dynamic in Africa. That would be interesting to hear at the same time. And the last question is, there was this association with water bodies, the presence of water bodies to the disease transmission, if you have found any association with that. Thank you.
I'm happy to tackle those questions. In response to the last question, we started looking in water, and the environmental signal led us to possums. So yes, water can be contaminated with Mycobacterium ulcerans, but it's possums that harbour the bacteria and shed it into the environment. And I don't think contamination of water helps explain the disease distribution that we see. In terms of the mosquito species, your first question, in that large survey we've just completed, one species was predominantly positive for Mycobacterium ulcerans. It's a species of Aedes mosquito called Aedes notoscriptus, and it was highly significantly positive for Mycobacterium ulcerans compared to any other mosquito species. In a previous study conducted not far away on another peninsula in our region, conducted by Paul Johnson and colleagues, they found several mosquito species positive. I'm not sure what to make of that, but we certainly observed a very clear association with one mosquito species in this study. And the explanation for that is intriguing, and we don't have the answer.
And I guess just to follow up before I come to Professor Makni, there's an online question for you, Tim, about the positivity rates that you get in PCR. I remember you showed out of 1,000 mosquitoes, you have seven coming up positive. But the person wants to know whether these positivity rates that you find in mosquitoes correlate with positivity rates in cases or patients that have Brule ulcer.
That's a hard question to answer. The positivity rate is around 0.5 to 1% of mosquitoes that we trap are positive for Mycobacterium ulcerans. We can show, and I showed in that spatial scanning study, that the areas where we have higher than background levels of positive mosquitoes overlaps where we have clusters of human Brule cases.
I guess just to summarize on that question further, they're trying to, or the one asking is trying to see if it's contamination or true causal correlation between positivity in mosquitoes, possums, and then humans as well. So maybe you can engage further on the Zoom because the questions keep coming back up there. So,
but we could just refer to previous published work that's shown that the further you move away from endemic areas, the less likely you are to find mosquitoes that are positive for Mycobacterium ulcerans. So there is a spatial relationship between mosquitoes with the bacteria and where the risk of Brule goes. Yeah.
Thank you. Professor Markin.
Thank you very much. My question also for our Australian colleagues. Thank you very much for the quality of your work. I have a lot of questions, but Daniel has already asked some of them. You didn't respond for the reservoir. If it's mosquitoes, we expect that there is more than one reservoir. Is there a study for other reservoir? And also for the transmission, you are thinking about excrement. Why it's not a blood bite meal? And the final question, sorry, it seems that it's a new epidemic in Australia, at least for recent years. Is there any ecological change in the possum population or something like this who explain these outbreaks? Thank you very much.
The interesting questions. Has there been a change? Not that we... Well, yes, the human population has expanded. The possum population has expanded. The bacteria has been introduced from the east of the state to these big urban centres in the last 20 years. Why, we don't know. So there have been some changes. I might just go to your question of alternate reservoirs, and just Dr. McNamara is going to present later, and we'll talk about alternate reservoirs that were discovered in a survey that she led. And what was your other question? You had one more.
The transmission, why you are thinking only for excrement and not blood meal, but blood meals for the mosquito, like leishmaniasis, for example?
I mean, what we don't know in great detail is how the mosquitoes are spreading the bacteria during blood feeding. Now, our proposition is it's a mechanical mode of transmission. This isn't we're not proposing the mosquitoes as biological vectors where there's a cycle of the pathogen in the insect, but there could be. This is mechanical. And I think the data I presented where we showed we had individual mosquitoes that had blood fed on humans and possums shows that there is a model of, say, the interrupted blood meal model where a mosquito might feed on an infected possum which has a Buruli lesion. So the mosquito becomes transiently contaminated with the bacteria which are abundant in the lesions, and then takes the second blood meal from the human. So I think that's my most plausible working hypothesis on the details of how transmission occurs. But it's a good question that needs some more research.
Yeah. I don't know whether, Paul, you want to add something to all these things, because it's Australia after all.
Thank you. Just to comment, Tim referred to, or one of the earlier questions was which species and how many species. And in that original work we did back, was published in 2007, there were 5 different species listed, but it included Aedes nodoscriptus, and in fact that had the highest maximum likelihood. It was just that work was dominated by this other species called Aedes camptorhynchus, which is a salt marsh mosquito, and that was so prevalent. Every time we got a trap, it was full of them, and they were positive at the rate of about 4 per 1,000. So it is very interesting to me observing when it's been repeated again on a different peninsula, which is only dozens of kilometres away, really, that you get this very focused thing. So there's plenty of negative mosquitoes in the Culex molestus group, for example, that are all negative for M. ulcerans, but it's the Aedes nodoscriptus that is positive. So we are starting again to think maybe there is something biological or behavioural about this particular mosquito. Thank you.
Thank you, Paul. Yes, I can't see your name, but yeah, the floor is yours.
Thank you. Andrew Steer, scabies guy from Australia. My question's for Steve. That was a great presentation. Thank you. Really interesting work and great progress. It's a quick question. Just around the kind of clinical impact of the model, are you, particularly with a skin model, just to hear your thoughts about scarring particularly for participants?
Thanks very much, Andrew. And I was very excited to hear your work on a Strep A human infection model. Look, presumably it will leave potentially a small scar, but we would the hypothesis is that it will generate, as Lydia was saying, maybe a small nodule to start off with, and then we'd treat it very early if it became an ulcer, or if it didn't ulcerate, we would treat it after a certain period of time, say 7 days, work in progress. If it didn't self-heal or if it failed to progress, it may well leave a small scar. We propose something similar to maybe a BCG scar on the arm, but instead of being on the arm, it would be on the back of the calf. There would be the option, presumably there'd be the option to surgically excise it to make sure that people have the optimal aesthetic outcome. I guess from a research perspective, the benefit of that is we get significantly more tissue for diagnostic purposes, but also to understand the local immunological processes that are going on, which is probably far more relevant to Buruli ulcer than what we might get from, harvesting serum or blood. But hopefully it would be a very small lesion, and that's the aim of the challenge model, is to generate something small and not rapidly progressive.
Thank you.
Thanks.
And Stephen, just to piggyback on that before I allow further questions, there are quite a few questions about the ethical implications of this model. And so can you kindly just shed some light on that?
Yeah, thanks. I didn't go into a lot of detail in terms of the ethical implications. We will have an ethicist involved in the research from the beginning as well. Obviously, we need to make sure that all participants are completely aware of the risks of the model and aware of the, potential... if it's a first-in-human model, then maybe there may be some unforeseen consequences. But at the very least, it needs to go through a Human Ethics Committee, and it has to have an independent committee to oversee the entire process from beginning to the end. We need to... I didn't go into the complex details of the eligibility criteria, but there's so many things to think about. Making sure that people are able to follow up with the protocol as well, so they don't leave with an ulcer that fails to heal or be treated. But yeah, there's lots to think about, but it wouldn't be done without careful oversight and approval. Thanks, Lydia.
Thank you for that response. Wow, I have a plethora of questions, but I'll go with yours first, and then the next three.
Hi, it's Rod Hay, and I guess it's for anyone who could answer this. My understanding is that there's a low level of infection, so the Buruli case is seen in other parts of Australia, like the Northern Territory. Is there anything that could be learned from those areas?
Paul wants to answer, so Paul, please.
That's a very good question. The conditions that we've described in our part of the world are present all the way along the eastern seaboard. New South Wales, Sydney, Brisbane, Queensland. You would expect M. ulcerans to be all the way up there, but it just isn't in great numbers. There are little focal spots. The most active other region is in far north Queensland, in the Mossman area, and it's an extremely small area that literally starts at the Mossman post office, like at a certain place in the town, and goes up to the Daintree River. And the population there, the permanent population, is about 3,500. And they have typically 0 to 1 to 2 cases per year. And they had this spectacular outbreak back in 2011 where they had 61 cases. The following year there were 11, and then they've gone back to 0 to 1 or 2. And there's an occasional case from near Darwin and an occasional case further down the coast, but it's like just a few. So what can we learn from that? Well, we've been up to that northern, far north Queensland area several times and sampled absolutely everything we could. And on one occasion, we got one extremely strong signal, and that actually was from the excreta of a bandicoot, which is a small Australian marsupial. It's a ground dweller. Got very excited by that, went flying back up there, got 250 more samples, and got another really strong positive, which was about 1 metre away from the first one several months later. So it was probably the other half of the excreta or something that was now in the soil. So, and that's been published, that little story. There's been some other researchers who've managed to get a few positive mosquitoes, but that's it. So that's very tantalising because the environmental conditions there and the climate is very similar to Africa. And to find one other example of the same sort of pattern that we see in Victoria is really intriguing. But if someone wants to have a really nice warm time in a nice part of the world working it all out, you'd be welcome to come and do it. Thanks.
Thank you. So, yes, you.
Okay. Merci. Ma première question était en rapport avec ce qui vient d'être dit. Ça veut dire, est-ce qu'ils ont eu une zone contrôle? Parce que le fait de concentrer la problématique sur une seule zone peut donner l'impression, les signes négatifs ailleurs pouvaient bien exclure cette hypothèse. Je voulais m'interroger sur pourquoi le hotspot de la...
I wanted to know why the hotspot that you shared with me on the map, which included no factors regarding opossums or insects, was not included. The map that was showed earlier, none of these elements were included. So I'm just trying to underline that there's a lot of work to be done still regarding what concerns the environment. And perhaps these teams that work with Mr. Gange can contribute to this work. Thank you.
So I'm sure this was going to Jean-Francois, who is not here. So possibly, if there's anyone from Jean-Francois' team who wants to attend. Okay. And Tim as well? Okay. Yeah,
I think I didn't use the translation service, so I'm going to... No, it's okay. You can correct me if I've misunderstood you. You wanted to know why we didn't provide more details in some of those maps on what was...
Yeah, the first one.
Microphone, please.
I see, without any risk factors. Look, I guess that was a summary, and the mosquitoes are everywhere. Those maps I showed with the pie charts were aggregations of 180 trap sites across 350 square metres of terrain, and the pie charts were just summarising areas, so the resolution wasn't there to show you the full distribution of our mapping or the granularity of the insect density across the area. But I guess rest assured, there are risk factors, as in mosquitoes, across the entire Buruli endemic area. Thank you.
Okay. Can't see your name, but yes.
Yeah, Sasha Knauf, FLI Germany. Congratulations to the Australian colleagues here, really great work. If you think ahead about interrupting transmission, one thing is, of course, controlling the reservoir system. And if you think about animal TB, then we have an example in Eurasian badgers and that transmit to livestock. Is there any idea about controlling Buruli ulcer in possums?
Yes, so we like the way you're thinking, and in the next session, Professor O'Brien will put forward a proposition in that regard.
Thank you very much. My question is to Professor Timothy Stenier. Thank you for the very interesting presentation. You mentioned in your presentation that Buruli ulcer is geographically very focal. So on the other side, we're talking about vectors, reservoirs, and some environmental correlations. Isn't it a little bit paradoxical because other diseases don't behave like this?
Paradoxical. I think our data explains the focal epidemiology we see quite well, in that you need that coalescence of the factors to come together. You need a high number of possums, you need a high number of mosquitoes, you need a high number of people, and then you need those possums to be infected with Mycobacterium ulcerans. And in Melbourne, not every possum is infected with Mycobacterium ulcerans. And we showed from our predictive maps that it's only in those areas where the possums are harbouring the bacteria that humans are getting the infection. So that explains the focal epidemiology we see. Maybe your question is perhaps also, how does it explain the focal epidemiology we see in West Africa? And that remains to be answered. Whenever we present a talk on our possums, the first question is, but excuse me, there are no possums in Africa. And I know Lydia and other colleagues are spending a lot of effort to try and find an analogous small mammal reservoir.
Thank you, Tim. I'll come to you, Ree, but there was a question with... yeah.
Thank you, Madam Chair. I'm Ronaldo Schulte from PAHO. My question is for Professor Steiner. You showed that possum excreta, and please correct if I got that wrong, could be a very powerful public health tool to identify human cases. I'm wondering if mosquitoes could be used like xenomonitoring to assist on that too. I don't know how easy it's to get the possum excretas, but mosquitoes, I think it's a bit easier, no? Thank you.
Yeah, I think either of those sample types could be useful for informing public health risk. I guess the possum's excreta is very easy to collect. Not to be too crude, but it kind of rains. Possums are arboreal, they live in the trees, and it rains possum excreta around our homes. So it's a very convenient analyte, and it's spatially trustworthy. Now, the mosquitoes we're talking about have a flight range of 2 to 3 to 4 kilometres, whereas the excreta has a range of about 100 to 200 metres. So it's abundant, it's relatively cheap to analyse in the lab, and it's spatially trustworthy. So my preference would be that it's the possum excreta that is the convenient environmental sample type to predict the location of future human Buruli risk.
And I think we'll just take one more question. I
was just going to say, but it is very boring to collect possum excreta.
Totally appreciated. Ree, I think you'd have the last one. And then if you have any further questions, please engage the speakers during the, when it ends, I guess.
Thank you. Today. Well, sorry. It looks like there's a good evidence that the mosquito is transmission in Buruli ulcer. But my question is, from the clinical point of view, Buruli ulcer is mostly like single lesions. But when I get bitten by the mosquitoes, it's multiple bites. So is there any explanation for that?
Yes, Paul wants to answer that.
I mean, I've got several patients who've got 3 consecutive lesions that are PCR positive. I don't know why you don't see that more often, but there's enough of it to be sure that that doesn't sort of, it doesn't falsify the hypothesis, I don't think.
Well, if you still have questions, please hold them till the end of the next session. I would like to invite us to all stand up for just a few 30-second health break and just, I guess, get blood flowing in your arms and do a quick turn if possible. I can't. Thank you all for indulging in that. So we'll start the next session now. So there are 4 talks in this next session, and if we stick to time, we can again start another round of discussions or end early. So I'm very happy to introduce the next presenter, Heather Jordan, who I've known since 2004, and she'll be telling us about some work that she's done in detection of Mycobacterium ulcerans among Aedes albopicus submerged from M. ulcerans contaminated water. So Heather, the floor is yours.
All right, thank you very much, Lydia. So there have been several mechanisms of emulcerans transmission that have been proposed, including exposure to emulcerans contaminated water or vegetation, or exposure to animal reservoirs or other excreta that have been shown to maintain emulcerans in the environment, or puncture of emulcerans contaminated skin, or insect vectors such as predaceous aquatic insects or mosquitoes, which is the focus of some data that I will present today. So evidence for the role of mosquitoes in transmission is strongest in Australia, and Tim and Paul did a beautiful job of explaining the role of mosquitoes in Australia thus far, so I'm not going to go into any detail with that. But there's less evidence in Africa where there's limited data of emulsirans positive field-collected mosquitoes. But in the laboratory, Wallace et al. Showed trophic transfer of emulsirans up the food chain and transstadial development of emulsirans only through larval instars. So they did not detect emulsirans in pupae or adult stages, although they did show that emulsirans could be transferred externally on bodies and legs of mosquitoes that were feeding on contaminated cotton balls. In another study by Wallace et al., they showed that mouse tails dipped in culture and bitten by mosquitoes could lead to Burley ulcer disease. And there are other studies that are shown up here, but because of time, I will not go into those. And these have all been really great studies, but there's always more work that could be done. So we were interested in mosquitoes being hatched and developed in emulsirans contaminated water in an effort to mimic a more natural environment for host microbe interactions. We were also interested in more transstadial studies using quantitative PCR and to understand microbiome differences, as there have been other mosquito disease systems showing that mosquito microbiomes may impact pathogen development and vector capacity. So our study objectives were to measure emulsirans presence and abundance in Aedes albopictus hatched and developed in emulsirans contaminated water, and to determine mosquito microbiome changes after emulsirans addition. So we chose Aedes albopictus because they have been identified in habitats that overlap with PCR-positive environments, such as rice fields or streams or drainage ditches. And these mosquitoes will breed in artificial containers, but they can also be found amongst dense vegetation. They are aggressive daytime biters and are vectors for a number of clinically relevant arboviruses. And interestingly, you can find the larvae floating upside down at the air-water interface using their siphons to breathe air. And in the laboratory, we can sometimes find emulsirans at the air-water interface as well. All right, so this is our experimental design. It's a little bit complicated. On the right-hand side is our experimental group, and on the left-hand side are the controls. We used GFP-labeled emulsirans, and we added the emulsirans to a container with DI water. Our control group was only DI water in a container. We used deoxygenized albopictus eggs, submerged those into either container, allowed them to hatch where we collected the larvae, and moved those into new containers with DI water. They then pupated. We collected those pupae. We moved those into new beakers with new DI water as well, put those into bug dorms, and allowed the mosquitoes to emerge. The mosquitoes were then aged for 5 days and allowed to blood feed, and the females oviposited onto egg paper. We then put those egg papers into new containers with only DI water, where they hatched, went through the developmental stages for generation 2 following these exact protocols. And then those adults were allowed to blood feed and lay eggs, and then we did the same thing where we put those eggs into clean water, allowed them to go through the generation, the developmental stages for generation 3. But I want to stress that emulsirans was only added in the first generation in that first container with those eggs there, so only one time. So we collected samples across all of these life stages. We also collected environmental samples, so water or swabs from the containers. And when possible, we collected casings from the larvae or the pupae as they went through their life stages. So we also had a subset of samples of larvae and adults that we dipped in bleach with additional water washes. And then those bleach adults were further dissected into head, thorax, or abdomen, to measure the internal microbiomes of these. So we isolated DNA and used quantitative PCR for presence and abundance of emulsirans, and we sent a subset off for 16S targeted metagenomic sequencing. And one thing I forgot to mention is that across both treatments and generations and life stages, we used completely new containers and equipment. Okay, so here are some of our data, and if you look at the bottom left, you can see that after we subtracted the autofluorescence coming off of the mosquitoes, we were able to detect some fluorescence amongst some of our larval, pupal, or adult samples, indicating that we did have some viable emulcerans amongst our samples, and these are actually among first generation. We didn't find any in our second generation or third. So on the top panels in A and B, this is presence and abundance, respectively, of emulcerans across life stages and generations. And what I hope you can see is that as you go from larvae pupae to adult, there is a decrease in presence and abundance of emulcerans as it is traveling through the life stages in generation 1. Also in generation 1, environmental samples and larval and pupae exuviae, or those casings, were also positive. In generation 2, we did have some adult samples that were positive along with larval exuviae. And then in the third generation, we only had one adult that was positive. Okay, so this is showing some significant microbiome data. And so in the top panel, we have significant differential abundances corrected for treatment of emulcerans versus control. So the numbers are basically showing indicator taxa of emulcerans exposure status across life stages. So you can see that there are 199 unique indicator taxa between larvae and pupae that then increase between the pupae and the adult, and even more so between the larvae and adult. And again, these are indicator taxa for emulcerans exposure status. And we weren't really surprised by this because one would expect microbiomes to shift as going through the life stages. But we did find some shared differential taxa as well that are across all of the life stages. So overall, these data do suggest that emulcerans may mediate differences between bacterial communities as mosquitoes progress through these life stages. In the bottom panel, we looked at exposed versus control larvae across generations. And you can see that there are 149 unique indicator taxa between control and the exposed group in generation 1. And then in the second generation, there were only 43 indicator taxa in generation 2 that did not overlap with generation 1. And then by the time we get to the third generation, there were no significant differences in relative abundance of taxa between control and the exposed group, suggesting that any emulcerans-mediated differences between these communities are less important as mosquitoes progress through the generations. All right, so quickly, I want to look at the internal body samples of larvae at the top and adult at the bottom. And this is, again, presence and abundance going left to right. So there was no significant difference between positivity and whole versus internal larvae, although there were significantly higher concentrations in the whole larvae. In the adult on the bottom panels, there was no difference in positivity or concentration between these different body types, although we did find positive samples among all the body types. Uh-oh. Okay, so again, these are some microbiome differences, and we'll first look at, Oh yeah, we'll first look at the larval samples. And so we're looking at relative abundance at the genus level of whole versus internal samples. For the top panel are exposed larvae and the bottom panels are control. And when we look within treatment, so looking first at the exposed larvae, we did find significant differences in microbial diversity between whole and internal samples and across generations. And the same was actually found when you look at the control samples as well. But we also did look, and I'm not showing the actual pretty picture here, but we did look at significant differences or measuring diversity between whole and whole, or internal versus internal, of the control versus exposed, so looking across treatments. And we did find a significant microbial diversity across treatments as well. And then when we look at the, well, just pretend it's working, and look at the adults, we find hang on. Yeah, there you go. We do find the same situation where within treatment we do find significant microbial diversity differences according to body part, but not by generation. The same in the controls as well. And then when we look across treatments, the whole versus whole show significant differences in diversity. Abdomen versus abdomen also significant differences, but not between head and thorax. But so we were interested in some of the significant taxa that we found. First of all, in the exposed larvae, we did find significantly more mycobacteria, which was, a win since we, had exposed them. And secondly, across all of the samples, we found significantly higher abundance of Wolbachia in the exposed group across all sample types. And then in the adult exposed, we found a higher relative abundance significantly of Elizabeth Kingia as well. And these were interesting to us because these are known insect, and particularly mosquito symbionts, that in other mosquito disease systems have shown to impact, again, pathogen development, abundance, and vector capacity, among some other things. Okay, so overall, our overall results are showing that presence and abundance of emulcerans was highest in that first larval generation and decreased as emulcerans moved through those life stages in both positivity and abundance. We did show that emulcerans was transstadially maintained all the way through to the adult life stage, but what remains to be seen is whether there was an increase in virulence from passage of emulcerans through the mosquitoes, and we also need to tease apart ratios of viable bacteria versus just detection of DNA. And unfortunately, after several attempts, we were unable to isolate emulcerans from these particular samples due to fungal contamination. We did show low percentage of adult mosquitoes positive within the second and third generations, but we also found environmental samples in larvae and pupae exuviae that were positive in the first and second generation as well. And what we think may be going on, although this does require more research, is that the females are inadvertently depositing emulcerans onto the egg paper as they oviposit. And there is precedent for this with other bacteria being deposited as during oviposition. Okay, we also found that that our data suggested emulcerans does shape microbiomes, although it did appear to be short-lived and was gone after the second generation. But whether this shaping was due specifically to emulcerans' presence or maybe to changes in mycolactone, or whether these taxa were simply covarying during cycling through another generation remains to be seen as well. But what does it mean with regards to transmission? Well, our data, at least in this system, don't seem to support that emulcerans is biologically maintained, although it does not rule out mechanical transmission, and it actually does suggest that emulcerans may be being dispersed by mosquitoes in this type of scenario. But when you think about dispersion of emulcerans, it's also interesting to think about dispersion or transmission of these other pathogens. As I mentioned, these are vectors for clinically important arboviruses, and so does the presence or absence of emulcerans that may shape these microbiomes alter transmission of these other pathogens or affect mosquito phenotypes that may impact vector capacity or behavior such as oviposition or blood feeding? Okay, quickly, we did have several limitations of this study, one being that we were the only 2 people in the building during the COVID shutdown. It was only 1 inoculum, single dose. We were unable, unfortunately, to isolate emulcerans due to some contamination and some other things as well. But in the future, we do want to use xenic mosquitoes and fiddle around with the microbiomes a little bit more, measure emulcerans viability and virulence, and also to look at what happens when emulcerans are exposure is constant as well. And with that, I would like to acknowledge my co-authors, particularly Jillian Masters, a former master's student in the lab who did the majority of the work, and my funding sources as well. So thank you.
Thank you, Heather, for speeding through that presentation. But, I mean, you raised some very important points. One of them being that you are, I guess, providing further evidence for this Barnett's model that we heard about earlier on from Tim's presentation on vector competency and the role of mosquitoes in the transmission of brucellosa. And it is also important that you are showing that the next generation of mosquitoes have to pick it up again from the environment that they are in, highlighting the importance of the One Health approach in understanding transmission of brucellosa. So thank you so much for that very insightful presentation. The next presentation is online. It will be delivered, hold up a second. Okay, so the next presentation is a skip. Elala. So we wait to figure out whether the presenter. Okay. So I think the online presentation will happen. It's a talk by Andrew Bulchin, and the title is Genomic Approaches to Predict the Origins of Borreliosis Cases. So Andrew, if you can hear us, the floor is yours.
Terrific. Just a quick sound test. Can you hear me? Hello, can you
Yes,
hear
we
me?
can hear you clearly.
Terrific. Excellent. Apologies for not being able to be there in person, but appreciate the efforts to make this work. So my name is, next slide, please. Next slide. So my name is Andrew Bulgins, and I'm a postdoctoral researcher investigating the genomes of Mycobacterium ulcerans in Southeast Australia. And on the top right-hand corner of this area is an image that depicts the sorts of environments in which we see Borreliosis transmission occurring in southeastern Australia and Victoria. And this is quite a contrast to the sorts of environments in which Borreliosis transmission is observed in the Northern Hemisphere.
Sorry,
and I mentioned
Andrew,
that I studied the genome.
Andrew, is it possible for you to turn off the video so that we can have a constant flow of the sound for now? Thank you.
Yep. I've just disabled the video. How's that now? Is that better?
Yes, it's better.
Okay. So basically, the genome is the entire DNA complement within a cell, and specifically my work has focused on the chromosome of Mycobacterium ulcerans, which is 5.6 million base pairs long. And we can use all of that information to tease apart bacterial genotypes at a very high resolution. And I've been looking at studying the genomes of Mycobacterium ulcerans for over 10 years now. And in that time, I was shown that the pathogen in the Australian region has evolved from a Papua New Guinea-like most recent common ancestor. And it's moved its way slowly south along the Australian eastern seaboard down into Victoria, likely arriving in the late 1700s. And this arrival date coincides with the movements of early European settlers in the region, suggesting that they may have played a role in the movement of this pathogen into Victoria. And once establishing in Victoria, it stayed rather quiet in the eastern part of the state, in Gippsland. And then, as Deb described on Monday, the pathogen, the disease began to spread westward. And we've been able to use these insights from genomics to show that the actual pathogen itself, the causative agent, has moved westward into regions of Western Port and Port Phillip Bay, establishing a number of well-defined endemic areas close to inner Melbourne City and Geelong. So our approach using genomics using the complete DNA sequence of Mycobacterium ulcerans has given us deep insights into how this bacterium has spread across the landscape. And we are now interested as to how this may be useful as a public health tool. So one of the key issues for epidemiologists is trying to work out where a patient acquired their Borreliosis infection. So with an incubation period of 4 to 5 months on average, it is very hard for patients to recall where they may have been infected with Mycobacterium ulcerans. So wouldn't it be great if we had a tool that could inform on the geographical origins of Borreliosis cases with high precision? Then maybe for once we could get ahead of the curve. Next slide, please. So the objective for this study is to develop a bioinformatics tool that can provide more accurate information on the locations where disease transmission is occurring. And this is rather important information because this can be used to direct public health responses to areas that are more risky for transmission. So, for example, through the use of a tool such as we've described, if an area was found to be most risky of transmission, then public health efforts could be directed to that area locally to disrupt transmission. So overall, the concept is quite simple. The input is a clinical isolate genome from a clinical lesion, and the output is the prediction, which is the likely location of acquisition in the form of latitude and longitude. Next slide, please. So while the concept is simple, the execution is reasonably sophisticated. We're using a program called Locator, which uses a neural network, which is a form of artificial intelligence that's modeled upon how the human brain works. And to teach this model about the diversity amongst these genomes, we've got a set of 345 clinical isolates with good epidemiological data. And we train the model using a process called cross-validation that ensures it's adequately trained and it's generalizable to new data. And then once that model has been trained, it's applied to make predictions upon a set of 168 testing set clinical isolate genomes. And the important thing here is that the model was trained and with a separate set, the 345, and it never encountered this testing set until after it was trained. And that goes back to that generalizability, how it behaves when it is encountering new data. So the issue with what I have just described is that if we were to build a model with the labels coming from the epidemiological investigations, that modelling approach would inherit the inaccuracies of the epi information that I've described owing to the lengthy incubation period of the disease. So to address this, we've recovered a panel of Mycobacterium ulcerans genomes from possum fecal material from the environment. So as Tim mentioned, possums play a major role in the ecology of Mycobacterium ulcerans in southeastern Australia, and we can detect the presence of this pathogen at quite high abundance in the fecal material in areas where disease transmission is occurring to humans. And the fortuitous thing about possums are that they are highly territorial and they have a homing range of approximately 100 to 200 metres, meaning that any insights from possum fecal material is likely to be very geographically trustworthy. And we can use the signals amongst those emulcerins recovered from those possum fecal excreta to geographically calibrate our training isolate set so that we can build models with higher confidence. Next slide, please. So what I'm going to show you now is the performance of this modelling approach when it's applied to that set of 168 test set isolates. And the way that we evaluate the error or the performance of this model is in distance. So it's the distance between the known origins, the latitude and longitude of the known origins versus the latitude and longitude that was predicted by the model. So that's depicted on the y-axis, and on the x-axis are all those predictions ordered from smallest to largest. And what's quite obvious here is that most of these predictions are actually quite low in their error. In fact, 77% of these predictions are within 10 kilometres or less of their known origins. 86% are within 20 kilometres or less, and 95% are within 30 kilometres or less. So overall, this approach has quite good predictive capacity. Next slide, please. So what does that look like on the landscape? Well, here's the map depicting the endemic areas that I presented in my opening. And basically, I've displayed here vectors that depict the error for all the predictions that were within 10 kilometres or less. So here, each line depicts, starts at the known origin of disease acquisition, and it ends at the predicted latitude and longitude provided by the model. And what you can see is that this resolution of error, it's quite obvious to discern where a clinical isolate was acquired, where that disease was likely acquired by that patient. And this is therefore a tool that has the potential to inform public health officials on where to quantify the amount of transmissions occurring between areas, and therefore identify more risky areas for transmissions of Buruli ulcer. Next slide, please. So how can we use this information in the real world? Well, here's an example. So in 2020, there were 19 human cases of Buruli ulcer in the suburb of Essendon, which is north of the metropolitan city of Melbourne. And it's an area where Buruli ulcer transmission has not been previously identified. And the health department were interested as to whether this may be local transmission occurring. So to address this, we conducted field surveys in Essendon and revealed that possum excreta collected from that local environment was positive for Mycobacterium ulcerans. And we were able to recover whole genomes from those possum fecal excreta and found that those genomes were identical to the clinical bacterial isolates from the patients from the Essendon area. So this consolidated evidence that there was in fact a new endemic area in Essendon. There was local transmission occurring in Essendon, and this led the Deputy Chief Health Officer, Professor Deb Friedman, to issue an advisory on behalf of the state government that was aimed to prevent transmission and improve disease management. Next slide, please. So further work for this study will likely involve the integration of many more emulcerins genomes recovered from environmental specimens into our analyses of many genomes from that part of southeastern Australia. So up until recently, all insights into the population structure of Mycobacterium ulcerans have virtually all come from clinical isolates or veterinary isolates of Buruli ulcer in animals, because it can be cultured from a lesion and therefore integrated easily into a whole genome sequencing workflow. But as we know, emulcerins is virtually impossible to culture from the environment. So now we've opened the door to include many more, to include emulcerins recovered from possum, for example, possum fecal pellets. And if we could include many of those into our phylogenetic trees, then we may be able to answer the question, is the population structure the same as what we see amongst the clinical isolates? And that could lead us to uncover more about the ecology of this pathogen. Now that we can recover emulcerins from the environment, its genome from the environment, we can also turn our attention to other positive environmental sources, such as mosquitoes from endemic areas, as Tim mentioned. And now that we can look at the various genotypes of emulcerins in the environment, we can track the spread of these genotypes in wildlife populations. Next slide, please. So to conclude, I'll just iterate that genomics is a very powerful tool, and it's informed us greatly on the historical movement of this pathogen in Australia. And we've shown that the genomic variation that we can recover can be useful to predict the origins of Buruli ulcer cases. And this is particularly useful because it's objective and accurate, and it doesn't rely on that patient recall. And the ability to recover emulcerins genomes from the environment could be quite useful to truth epidemiological insights, and particularly provide feedback to epidemiologists working with Buruli ulcer. And overall, an approach such as that we've described here today has the potential to improve disease management and reduce the incidence of Buruli ulcer. Specifically, identifying where transmissions are occurring could be very useful when designing interventions, and overall will allow us to have a deeper insight into this pathogen's ecology. Next slide, please. It leaves me with my acknowledgement slide. I'd like to thank a number of people here who have made this work possible, particularly Tim Stanier, who's taken me on as an honours student 10 years ago now, and then kept me on as a PhD student and now as a postdoc. And it's been a terrific journey. In fact, I was at this meeting in 2015 presenting at the early stage of my PhD, and it's a pleasure to be back giving an update as a postdoc. I'd like to thank those at the Microbiology Diagnostic Unit for providing sequencing and bioinformatics support, and Elaine Tay from the Department of Health, who's provided us with the epidemiological information to make this work possible. Also, Koen van Delanutte from the Institute Pasteur in Cambridge, who was previously in the SNEER lab. He was instrumental with the fecal surveys at large scale, providing us with the material to then recover genomes from. Paul Johnson, of course, and a number of people from the Victorian Infectious Disease Reference Laboratory who have provided us with the clinical isolates to allow us to do this whole genome sequencing. Thank you.
Thank you, Andrew, and for finishing with 2 minutes or so to spare. And your presentation really highlights why genomic epidemiology is really important in transmission studies. I think it's even more applicable now where sequencing is much cheaper than it used to be before. And we have point-of-care sequencing techniques like the Oxford Nanopore platforms. It will be interesting to see how you were able to retrieve the MU genomes from the possum excreta during the discussion. So that's one of the questions that I guess we should start thinking, or you should start thinking about answering. The next presenter is Bridget McNamara, who is going to give us a talk on environmental risk factors associated with the presence of Mycobacterium ulcerans in Victoria, Australia. So Bridget, there you are.
Thank you, Lydia, and thank you to the organisers for the invitation to present today. So as Lydia mentioned, I work at the Barwon Southwest Public Health Unit, which is as an epidemiologist, one of the people that Michael was just talking about in his presentation, and we'd love to use that work to look at the truth of insights. So the work I will present today is work that's been done on environmental surveys examining the presence of Mycobacterium ulcerans in southeastern Australia. And at the beginning, I'd like to first acknowledge the work of Kim Blasdell, who's pictured in the top left of my slide, that's now there, who is the lead author on this study, as well as my study co-authors listed there. Thank you. Yes, thanks. So Buruli ulcer, caused by the environmental pathogen Mycobacterium ulcerans, is notifiable in Victoria. And as we've heard from Deb Friedemann and many others today, we're seeing an increasing number of cases in Victoria, as well as expansion of the endemic areas to new geographic locations. And you can see on the graph that we've seen a few versions of today that the peak of cases was last year in 2022 with 342 cases. And that the endemic areas have mostly been coastal areas surrounding Port Phillip Bay in recent times, which are key holiday spots in Melbourne, so a lot of visitors. However, in recent years, there's been new urban environments that we've heard that are non-coastal in inner Melbourne and also inner Geelong. So as we've heard from Jean-François earlier, it's important to understand how Mycobacterium ulcerans is circulating in the environment and how and to inform transmission pathways. Next, please. Thank you. So the aims of our study was to examine the presence of Mycobacterium ulcerans in residential properties across endemic areas in Victoria, and specifically to look to determine which environmental samples were most likely to contain M. ulcerans DNA, as well as viable M. ulcerans bacteria. We also aimed to more broadly identify environmental features that were associated with M. ulcerans detection at the properties, as well as human cases at the property. So we conducted environmental surveys, which were conducted as a case control, as a subset of a larger case control that recruited 283 laboratory-confirmed Buruli ulcer cases notified between June 2018 and June 2020, as well as 520 postcode match controls. And these participants were sent a questionnaire, one question on which was, would they be willing to have an environmental survey conducted at their property? And so we had a high response rate to this, so over 90% of the cases and over 88% of the controls were willing to have environmental surveys conducted at their properties. So from this, 115 case properties were purposefully selected to have a range of properties across the endemic areas, as well as then randomly selecting 115 postcode match control properties And these were properties that had no household members that had ever had Buruli ulcer. We also did a small number of repeat surveys, so 27 repeat surveys were conducted between 3 and 9 months after the initial survey to look at how the status of the property changed over time. So before the field researchers attended the property, a property outline was created which had the locations of the buildings on the property as well as large environmental features on the property. And then also the altitude and property size were noted. At the property, a maximum of 20 samples were collected. So when the field researchers arrived at the property, they would look at the type of garden features, so whether the garden was native, mixed or non-native vegetation, as well as noting other features such as overhead power lines. And then noting the location of the 20 samples collected. So the properties had 2 soil samples, one from either end of the property, and then if bore water was present, a single bore water sample was taken. And for the remainder of the samples, this was done in a stratified way to sample the representative features of the property. So we collected plants, water, soil, mammalian feces, and insect samples. The detection of the M. ulcerans bacteria was by PCR in a staged process. So we conducted a screening PCR, which looked at the detection of mycolactin-producing mycobacteria, DNA. And then secondly, confirmatory PCR, which is more specific for the M. ulcerans bacteria. And finally, a viability assay looking at ribosomal RNA was conducted to look at whether the presence of viable bacteria in the samples. For each, the proportion of each sample type that was positive at each level was noted, as well as a status for the property if at least one of the sample levels was positive. And you can see in the map here that which is an overview of where the surveys were conducted, that we had surveys across the range of endemic areas in Victoria, with higher numbers in the areas where we had the highest case incidents. So in the Mornington Peninsula, the Bellarine Peninsula and Bayside suburbs. We found that Mycobacterium ulcerans was more likely to be detected and also confirmed in case properties than control properties. And the map down below shows where we found the different status properties. So you can see in the dark brown, it shows where the properties with viable bacteria were detected. And this correlates well with the high incidence areas. Yes, please, next. Yeah, thanks. So in total, we sampled over 4,300 samples that were tested, and 11%, we found an 11% positivity rate overall. We found that faeces samples were most likely to have the bacteria detected, so in 20% of faeces samples, and this was also 30% of fox samples and 24% of ringtail possum samples. You can see in the next column that the confirmatory PCRs showed that similar results to the screening one, except at lower proportions. We also found M. ulcerans detected in the soil, as well as in plants, water, and in mosquito. In total, 1.5% of the samples were viable, contained viable bacteria detected on the viability assay, and all of these were faeces samples, so ringtail possums, brushtails, and fox. We looked at whether in the repeat surveys, whether the status of the property changed between the 2 time points, and we found for the majority the status was unchanged. So positive properties remain positive or negative remain negative. However, there were a small number of properties that changed status between the 2 surveys, and so going from positive to negative in 6 out of the 27 properties, or negative to positive in 2 of the 27. And one of these properties that became positive in between the two surveys, we noted that there was land clearing at an adjacent block, and it was hypothesised that infected animals may have moved from this block onto the property during that time. So we examined environmental features more broadly that were associated with positive detection of emulserins using univariate and multivariable logistic regression models. And what we found were that MU-positive properties were more likely to contain native vegetation, especially moonas and coastal tea trees, which are two species that are common to Indigenous areas, as well as more likely to have ringtail possums present at the property, overhead power lines, to have more saline soil and alkaline soil, and for the properties to be larger and at lower altitudes. They were less likely to contain non-native species, so this is just one species listed there, the Pittosporum species, and less likely to contain brush-tail possums. Whereas case properties, as I mentioned, were more likely to have positive samples at the property than control properties, and particularly positive mammalian faeces samples. They were also more likely to have specifically ringtail possum faeces present. Oh, sorry. Thank you. They were also, case properties were also more likely to have overhead power lines. So this is one thing that possums use often to get from property to property, climbing along the wires. However, when we controlled for the presence of possums, this was still a factor. So it's possible it could represent an unassessed environmental factor in terms of age of the area and growth of vegetation, or still be reflecting the more connected possum populations. We found that case properties were less likely to have moonas at their property. So, and we also, the field researchers noted that there were garden modifications at case properties. So cases appear to have modified the garden, potentially cutting down these native trees due to the hypothesised relationship between these trees and their also acquisition. So we conducted, there are a few limitations. We conducted the surveys at residential properties only. However, this was by design because we wanted to look at the hypothesised acquisition at residential properties. However, it does mean that if there were environmental features that were just outside the property, such as water bodies, that these are not represented in this study. We had limitations on the number of samples per property. However, 20 samples was sufficient for most properties, and only particularly large or complex properties may have had been undersampled by this. As I mentioned, the repeat surveys were, I think I forgot to mention, but the repeat surveys were limited by COVID-19 pandemic, so the timing was affected by the travel and work restrictions that were in place in Victoria at the time. And finally, the viability assay is relatively new in the environmental samples, and so we saw the most viable bacteria detected in samples that had the highest bacterial loads. So there's possibly more field validation to check whether samples with lower bacterial loads would also show as viable. Thanks. So in summary, we found that MU is common in endemic areas of Victoria and more common among case properties. And the findings of the extent of MU and viable MU, so emulserins, in mammalian faeces, especially in ringtail possum faeces, strongly support the role of this mammalian species in circulating and concentrating the bacteria in the environment, as well as supporting possible zoonotic transmission in the pathways for VU. The novel finding of viable MU in fox faeces suggests a potentially previously unrecognised role for this species in circulating the environment, the bacteria in the environment, as foxes travel larger distances than possums and could possibly contribute to the spread of the emulserins to new environments. The associations that we found with the broader environmental features tended to be features supporting possum food sources, habitat and mobility. However, we also found emulserins DNA in other samples, so in soil, plants, water and in mosquito. So these sources are also may be important. And I will note that the number of insects in this, collected in this survey were relatively small. Finally, emulserins is mostly, but not always, persists over time at the property. So there is some variation in risk in these areas. We did find that properties that were most likely to remain positive is when there were positive mammalian faeces at the property. And so to key public health messages that arise just from the data that I've shown you today, in terms of the first is to avoid direct contact with possum faeces or soil contaminated with possum faeces in activities such as gardening or occupational exposure where there could be a risk of puncturing injury. This study and also the broader case control study, which we have presented as a poster at this meeting, have shown that skin hygiene and protection appear important to mitigating the risk for Buruli ulcer and in the protection from injury as well as mosquito bites. As outlined in our poster, key preventative measures were that immediately washing and treating any wounds obtained outdoors by stopping activity to do so, particularly puncturing wounds, and also using insect repellent, as well as wearing clothing covering both arms and legs. And we found that there appeared to be a cumulative effect with lower odds in those reporting more of these behaviours. And just finish by saying that the importance of possums in circulating Mycobacterium ulcerans suggests that a better understanding of the possum locations of infected possums through the possum excreta surveys, as others have mentioned, as well as interventions to reduce the infections in possum populations are likely to inform prevention efforts. And my acknowledgements has gone, but I would like to acknowledge the Beating Buruli in Victoria research partnership and our funding sources, as well as the participants at the property. Thank you.
Thank you, Bridget, for that presentation. It brings to light the huge discussion, I call it the elephant in the room, about using or relying heavily on DNA in transmission studies. It just creates this web of organisms that are positive. And I think going forward, we have to start thinking about innovative ways of simplifying the web, you know, and really pointing our finger really at one organism. Thank you so much for that presentation. So finally, we have the last presentation for this session, which will be given by Danielle O'Brien. And the title is BCG Vaccination of Possums Against Mycobacterium ulcerans. So Daniel, the floor is yours.
Thanks, Lydia, and just move that. Thank the Scientific Committee for allowing me this opportunity to talk, and also hopefully some of you are still awake, and sorry for keeping you from leaving, but I will run over a bit of information that's already been said this afternoon. But what I plan to do is present on behalf of my colleagues from Barwon Health and CSIRO in Geelong, but also from the Doherty Institute in Melbourne, and also in our near neighbours in New Zealand, a proposed project to look at the oral bait BC vaccination of the Australian marsupial possum, which you've heard a lot about today, against Mycobacterium ulcerans, in a hope that we can interrupt the transmission of MU and do something about what you've already heard is a worsening epidemic in Victoria. So again, this has been shown a lot, but I just want to emphasise the three things that's why the epidemic's getting worse. First, we had our most cases ever last year, and they were 5 times more than they were 10 years previously. Second of all, you can see on the slide to the right where the endemic areas were just 5 years ago. However, since that time, we have new endemic areas in the Surf Coast, down by the beach, and in the urban centres of Geelong and Melbourne, as you've once again heard. But thirdly, we've also been noting a high proportion of severe disease, so that prior to 2010, about 20% of cases were categorised as WHO stage 2 or 3, but post-2010, we've seen a high proportion at about 30%. So a really important breakthrough you've heard about many times today was by Janet Fife and their colleagues Tim and Paul, who are in the audience today, that basically identified an outbreak in Port Lonsdale in the early 2000s, the fact that possum excreta were DNA positive for Mycobacterium ulcerans. but also when they captured possums that they did not just have positive faeces, but many of them had clinical ulcerative lesions, especially on the tail, face or on the paws. And further work from their group has also shown that those isolates from possums are genetically indistinguishable from those from Victorian human patients. Tim's already shown you, but I'll just reinforce that information, that there's a very strong co-location between where you find the positive faecal samples and where you find human cases. So in this example from their team in the Morning Peninsula, you can see the fecal pellets in the small oval dots, and those that are black are negative, but those that are red are positive, and you can see that the red positive samples are very closely located to the human cases, which are the, I'm not sure why they're happy, but happy yellow faces. But just another example from our own region where we live and work in Geelong is we've done 3 prospective samples in 2019, 2020 and 2022, and this is what you can see on the right of the slide, but also looking at what's happened with Buruli ulcer notified cases in our region. And I just want to emphasise that here in 2020 we found a positive faeces in what's called the Newtown area, and then within a year we started to see human cases developing. Likewise, in Belmont, we saw in around about 2020 possum faeces that were positive, and then we started to see 2021 onwards human cases. The same pattern repeated now out in the suburb of Highton and the inner suburb of Marshall. So once again, very strong co-location. In fact, often when you go down a street collecting possum samples, the ones that are positive are right outside the houses of the human cases, and they may be negative down the street. Furthermore, our group led by Dr. McNamara, who's just presented the environmental things that we, in a large case control study between 2018 and 2020 involving nearly 300 cases and more than 500 controls, that people who reported possums on their property had a 5 times higher risk of developing Buruli ulcer. And not only that, the more possums they reported on their property, the higher the risk of them developing disease. I'd just like you to note the green circle slipped, but I wanted to highlight the BCG vaccination was associated with a 40% reduction of risk for Buruli ulcer also in this study. And Bridges, of course, just presented the environmental component of that study, but I just wanted to reinforce the fact that, firstly, that again, the strong association between positive faecal samples and possums, but also that the RNA testing suggested that many of them were viable and therefore potentially involved in transmission, but also again that environmental features associated with possums, such as possum food sources, their habitat or the mobility, also were associated with increased risk of Buruli ulcer. And then finally, in a case report from last year that Professor Johnsons and Stenier are also involved in, we actually showed evidence of direct transmission from a possum to a human in a patient who went to tend an ailing possum, was bitten on the index finger, and within 6 months developed a Buruli ulcer exactly at that site, showing that it is potentially to transmit directly from a possum to a human. So I think the evidence, as we heard a lot today, is pretty strong for the association that possums are involved in the epidemiology and the transmission of Buruli ulcer. You've got the very close co-location of cases, the fact that you can predict with positive possum faeces, the fact that cases will develop in an area, through case control and environmental surveys showing a strong association between possums and Buruli ulcer cases, the fact that when you test the possum faeces, they do appear to have viable organisms, and also that we have shown that you can directly transmit from a possum to a human. So using this information, what can we do to try and impact what, as you've heard many times, is a worsening epidemic of Buruli ulcer in Victoria? Well, one thing is to increase public awareness of the fact and trying to discourage contact with possums, such as don't touch diseased ones or wash your hands after gardening. But in fact, our possums are very well adapted to our urban and suburban settings. as Tim's already alluded to and Paul, the feces are very, almost ubiquitous around our gardens, and so actually limiting contact is in fact very difficult. Culling, of course, is another option, but it's not really an option for us because possums are a protected species, so that is forbidden by law, but also they play a very important part in our ecosystems, and there could be significant unintended consequences from removing them from the environment. And furthermore, I suspect there would be significant public discontent with that method. So we really need something that addresses the possum problem, but in an innovative way. And so we've developed the concept of vaccinating possums with an oral bait BCG vaccine that will hopefully reduce the actual prevalence of disease in the possum population and hence reduce the number of cases in the human population. But I'd like to point out that in fact, it has been mentioned, but most possums probably die of this disease, and it's quite a slow and painful demise. And in fact, this would also be protecting the possum populations from their own morbidity and mortality. So what we aim to do is develop the tools for a non-destructive method of managing a zoonotic pathogen in its reservoir host that will have human and animal health benefits without damaging the local ecosystem. So why the oral bait BCG vaccination idea? Well, in fact, oral bait's been used very successfully in a number of diseases to vaccinate wildlife hosts, such as the rabies virus in Eastern Europe and the swine fever virus in Europe as well. But further to that, as was mentioned earlier, actually oral bait BCG vaccine has been used successfully to protect wildlife reservoirs against bovine tuberculosis in such things as the badgers in the UK, in deer in North America, and in boars in Spain. And furthermore, and most importantly, our colleague on this, Bruce Buddle, and his team in New Zealand, where they have Australian brushtail possums that are actually a pest species but also carry bovine tuberculosis, and that is then infecting cattle and potentially humans. They have shown that with the use of an oral bait BCG vaccine that you can reduce, well, you can produce immunity in these possum species and reduce their risk of disease by up to 95%. And this will last for at least 2 years. And furthermore, it's not just in the laboratory they've shown this, they've actually shown it in field trials that it is effective in reducing the prevalence of bovine tuberculosis in their possum species. But will oral bait BCG vaccine work against Buruli ulcer? It might work against bovine tuberculosis, but the next thing is, will it work against Buruli ulcer? Well, there have been 2 large randomised controlled trials of BCG vaccination for the prevention of Buruli ulcer disease conducted in Uganda in the late 60s and early 1970s, and both studies were consistent with BCG producing a significant but only short-lasting protection against Buruli ulcer disease. You can also show that mice vaccinated with the BCG vaccine will get protection against Mycobacterium ulcerans disease. And as I pointed out earlier in our case control study, we did find that a history of BCG vaccination did reduce your risk of Buruli ulcer by 40%. Oops. So with that in mind, what we've proposed is a 4-stage project. Starting with the development of Mycobacterium ulcerans common ringtail possum infection model, and that's where our colleague Steve Mewey comes in. You heard about him producing his Mycobacterium ulcerans. Well, we would be using that to inoculate subcutaneously possums either in low dose or high dose with Mycobacterium ulcerans, and seeing whether we can establish a disease model in the possums, monitoring both their excreta but also their clinical signs. If this is successful, then we would aim to test BCG vaccine administered, again inoculated, not orally, against MU in the common ringtail possums with controls and challenge groups, and measuring their effect on clinical disease, but also measuring the immune response through blood samples. Once again, if this is effective, to then see if we can develop a palatable and attractive oral bait for the delivery of BCG vaccines. Now, this has been done in New Zealand in brush-tailed possums, but we're dealing here with ring-tailed possums. I should have mentioned we're using ringtails because they have the highest prevalence of disease compared to brush-tails, but to see if we can develop something that will be palatably attractive. And if that works, to then test the oral-baked BCG vaccine against the M. Mycobacterium ulcerans in these ringtail possums in the in-house model, but also if that works in the natural field settings. So where are we up to? So far the team at CSIRO have actually established a successful housing and husbandry conditions within their facility for ringtail possums. We are in the process of collecting 6 ringtail possums to start with the Mycobacterium ulcerans challenge beginning in a few weeks. I did very generously donate the possum that keeps me up every night in my roof, but once it was caught, it was tested for Mycobacterium ulcerans, and I'm sad to report it was positive, so it's been released back into my house. So now I still have the possum, and now I've got a possum with M. ulcerans. So the trials and tribulations of research. But anyway, that's next step starting. So what I hopefully will be able to do in the next meeting is present you some further positive news as to how this project's going, so that when you bring your surfboard to our area, your risk of acquiring Buruli ulcer will be significantly less than it is at the moment, which is small, by the way. Thank you all.
Thank you, Dan, for that presentation. I can't wait for 2 years, the next 2 years, to see what the outcome of the study would be. The question that I would have, though, is, in Africa, what do we do? Because, again, we don't have the possums. So I'm wondering what we'll be vaccinating there. But it's more room for work for us. Okay, so we are at the end of this afternoon session and at the beginning of our discussions. So I would open the floor now. If there are any questions, I would. Okay, so Professor Monkney, sorry.
Thank you very much. I want to thank our Australian colleague for this high information and outstanding on Buruli ulcer in Australia. But maybe my question is not only for them, it's maybe for the African expert of Buruli ulcer. What's the lesson learned from Australia to Africa? Kingsley, I think it's for you.
Well, I'm not involved in Buruli transmission, but I was going to make an analogy, so I use this opportunity to do so. In Africa, we said that, or in neglected diseases, we say that they affect the poor. In Africa, most of the endemic areas are also in the very poor areas. The Australians, they have not so far provided information on demographics, economics of people who are affected. And it would be nice, that's the analogy and comparison I would like to hear from the Australian colleagues. Yeah.
Paul.
Thanks for that question, Kingsley. We don't have systematic data. However, many of the people who are cases have enough money to have 2 houses because that's their holiday houses down on the Mornington Peninsula and the Bellarine Peninsula. So it's actually the higher income group most at risk. Thanks.
And I guess due to some of the years that I've had in transmission research in Ghana and in Cote d'Ivoire, I can say that we've looked in fecal matter of trapped rats as well. We've been able to detect emulsions in some of these fecal matter. We've also been able to detect lesions on some of these trapped rodents. Most of them are Mastomys species. And in these lesions, you can get PCR positive for Mycobacterium ulcerans. What really hasn't been done is a prolonged study in, should I say, systematic matter to pinpoint one particular rodent or one particular organism. We've also looked at trapped mosquitoes in Africa, but we don't see the same numbers that the Australian group sees. And it again raises questions about the strain of ulcerans that you have circulating in Africa versus Australia, and more importantly, the congener of mycolactum that they make, because that can also have implications for both vector competency studies and virulence and pathogenicity. So there is still room for more research, particularly in transmission in Africa, and obviously we need funding to continue this work. So yes.
I'm going to make a question, but in Spanish, because Spanish hasn't been spoken here in this meeting yet. So, la pregunta es...
Yes, the question is, the Buruli ulcer seems to not be a big problem in the region of the Americas. Of course, there are some cases that have been diagnosed positive, but this hasn't been such a prevalent issue as it is in Australia or in Africa. Some of you that have presented these very interesting presentations, I would like to ask you a question. Do you have a hypothesis of why this is the case, that is not as prevalent in the Americas as it is in Australia and in other countries? Thank you.
Okay, so I think the question is asking whether, do you have an idea why the prevalence is so much higher in some of the places that you've looked at in Australia compared to some of the South American countries?
And why is it not that much of a problem in the Americas region, apparently, as it is in Africa or in Australia?
Yeah. So any of the presenters from the first session, or Daniel? Yeah, thank you.
I was just going to say, I mean, I think what's really driving our epidemic at the moment is the possum infestation. So we have a different situation where we have an identified mammal that's driving it, and there may be others as well, but I suspect that that's the major difference. And maybe in these other areas you don't have something that amplifies and increases the risk like we do in our environments.
But I guess it also raises a question about the foxes that are now coming up. That's another complication in the web, you know, so how about the foxes?
Yeah, no, so absolutely. So we're in the process now of planning further research because, I mean, at the end of the day, the possums was found initially, it was a surprise, and so part of the reason for doing the whole environmental study was to look and see were there other potential sources. And I think the fox, when we initially thought about it, thought, oh, well, that's just because they eat possums probably. But in fact, their feces were viable when tested. And one of the things that hasn't been solved is why does the disease suddenly appear in a new area? Now, one of our theories, not just me but others, is that possums, they're actually very territorial, but they do get moved because you're not allowed to kill them, basically. People catch them and take them and let them into someone else's house, but often a long way away. That maybe that was doing, but it's possible that a fox that does roam, you know, a long way could be part of the puzzle. So we're definitely looking into that further, but we don't have anything on that at the moment.
Thank you, Daniel. Ree, was your hand up or was somebody else? I saw a hand. Oh, Paul, yeah.
Yeah, just picking up on your point, you could equally say, why is it only in these little parts of Australia? Because the conditions for it are widely spread. And then if you say it's widely spread in Australia, you just have to go back 50 years and it wasn't. So it's a rapidly changing situation. And I would wager that if you introduced or somehow emulcerans, the organism arrived near Sydney or Brisbane, the same thing would happen. And same thing could happen in the Americas as well if it somehow got there, and maybe it's happening in Japan. We saw that. It seems to be new and it could expand there too.
Thank you. Yes. Yeah.
I'm Ciro from the Brazilian Ministry of Health. I believe we have 2 reported cases published of and ulcers in Brazil. And I think the study that we recently made on those cases may be a clue for that. One of them, the patient had traveled for Brazil, Peru, and possibly to the northeast of the South American continent where the disease is endemic. And we believe that it was not an autochthonous case. And the second one, we, together with the WHO, by the time, with the aid of Professor François Postals and Professor Roger Pradino, tried to retrieve the sample, but the authors never sent the sample for re-identification. So we believe that was not actual M. ulcerans. But Brazil has the right environment for the bacteria, and we don't know why we don't have. Maybe this theory and facts of vector-borne disease may be a clue, because Brazil is not a very attractive country for migration, especially for language, where this disease is endemic. But Australia is a developed country, is more attractive, and has the right environment. That's what we know from Brazil. Thank you.
Yeah, thank you.
I just want to add that also in Japan, we have bloody ulcer cases, and to date, we've been diagnosing a total of 83 cases, which are very sporadic in distribution, and we don't know how these cases are transmitted. And it's ongoing studies, but we don't yet know. So just to add to the discussion of where the diseases are. Thank you.
Professor Phillips.
All right, thank you very much. I think the more I hear the story from Australia, the more headaches I have, because I've been trying to think about what the story should be from Africa. What we noticed, I mean, for the several years we worked on Buruli, was the fact that these patients were coming from particular areas. If you look at the distribution in Ghana and some of the West African areas, they come and the patients live around particular rivers. And that was so clear. The only thing was how to associate the rivers. What was it? Was it an insect? What was it that was linking the patients to those areas? And at least those were the patterns that we see. If you asked me to mention 5 rivers that those patients seem to live around, or their communities are spread around, it's very easy to say. The only thing is we've not been able to find the link, exact link. So, I mean, we don't have possums, but maybe we need to be looking for other things that will link these areas to the patients. Thank you.
Thank you, Richard. There's a question, yeah, to my left.
Sorry, my question is to Daniel O'Brien. While you were speaking, I was thinking through the implications that what you were saying might have for 9-banded armadillos in the Americas. I'm sure you're aware that they have been implicated in transmitting Mycobacterium leprae in the southern United States, and it is known that there are M. leprae infected armadillos also throughout other countries of Latin America. And in fact, I believe that I'm no authority on that topic, by the way, but that they are spreading. And also that the leprosy infection inside the armadillo population in the United States is spreading. And I wasn't aware that, say, BCG, oral BCG was available. And so I was wondering whether that would have implications for possible public health measures towards armadillos.
Yeah, thanks. That's a very interesting point. I'm no expert on armadillos either, but what I do understand is that BCG does have protection against leprosy. So it would be a bit the same as we're hoping with the M. ulcerans. I'm not sure how available it is. I mean, I know they've used it in these wildlife programs, but on what scale and where they get it from, I'm not aware of that. But I think it's a very interesting idea, yeah.
So we have one question here, and then I'll take a few from the online platform.
Thanks. Is Andrew still online after his talk? Is he on the call?
Andrew, are you still online?
Yep, still here.
Yeah, hi Andrew.
Yep.
It's Eugene from Australia. My question is that as the database builds with your genomic approach to predicting risk, will that give you better discrimination to identify potential new clusters or cases, and perhaps using the RNA assay that Bridget described, which talks about viability, might even give you another level of refinement. Can you perhaps comment on that approach?
Yeah, so with anything that's machine learning and training, generally more data is better. We'd certainly need to continue monitoring the genotypes in the environment as the pathogen continues to perhaps spread throughout Victoria and other regions, so we need to keep an update database. In terms of viability, yes, that's also a key question. Are these actually in the environment? Do they have the potential, the capacity to cause disease? Are they viable? Yep.
So Andrew, whilst you're still online, could you just in a few minutes tell us about how easy it was to sequence and assemble the genome of MU from the fecal matter?
Sure. So basically what we did was we constructed a set of RNA baits that had complementarity to the emulsiran's chromosome from the Victorian isolate. And these are genome-wide baits that have the ability to allow us to perform targeted enrichment. So we could essentially extract genomic DNA from a possum fecal pellet, mix it with these RNA baits. They have complementarity, they bind, and they're biotinylated, so we can then separate them out from that solution. And then essentially we prepare library prep as most conventional whole genome sequencing practices and sequence them with Illumina short read technology. What we did find was that there was quite a strong correlation between the input CT, so the IS2404 assay, lower CTs were better, therefore higher abundance of the genome is better, which is quite obvious. And as you have higher CTs and less abundance of the genome, the sequence capture performance was reduced. In Victoria, we had a number of key single nucleotide polymorphism sites that we used to build what we call phylogenomic trees and to do other typing practices. So we were able to specifically build this set of RNA baits around our key markers to enhance detection for those areas.
Thank you. I have two online questions for either Tim or Paul. Is there a correlation between infected mosquitoes, Brucellose cases in the light of climate change? That's one. And then the next one is, could flies be a vector for Brucellose? I guess that one is for everyone else.
I think the first question is a good one, but I don't have an answer to that, and I'd offer that to any of the colleagues here might want to comment. But possibly, climate change is affecting all of us. And then the second question was the flies. Yes, well, we actually, because possums get lesions, possums die from this infection. The possum carcass, we've tested flies, the larvae of flies, adult flies. Some of them are emulsirins positive. Are they spreading it to humans? Probably not. Are they spreading it in the environment? Perhaps. So certainly there's, flies do have a role here in some capacity.
So I'm sure there are more questions that people want to engage the speakers in. However, it's exactly 5 o'clock and I'm going strictly by the schedule in front of me. I want to take a moment to appreciate all the speakers. Thank you so much for sharing your work with us. It was really interesting. My recurring challenge with the transmission section, the session at Brucellose meetings is you leave more confused sometimes than with answers. But again, it just means there's more room for research. So, Kingsley, thank you.
All right, thank you. very much, Lydia, for chairing this session. Before we close, I have a few announcements, but I also have one question for the Australians before we finish transmission discussions. In different meetings, people, when they hear, oh, there is borreliosis in Australia, wow. Do they affect the Aborigines? Or, so please answer this question in this meeting so that nobody will ask me these questions again. Okay.
May Smith, you've met May Smith, who for a long time was a very careful documenter of what was going on in her area. Yes, there were some Aboriginal people who got the infection, but at no higher or lower rate than you would expect by population. So we've seen it in people of all ages, both all genders, all races. It's just a probability function of where you are. If you don't go to an endemic area, you cannot get it whoever you are. And if you do go, you can get it within 1 hour of being there. That's the shortest we've found. And then leave and start seeing something happening 4.5 to 5 months later on average.
All right. So, a few remarks and reminders. Dr. Kazim, who is the lead person for neglected tropical diseases at the WHO country office, regional office in Manila, would like to have a side meeting with all the countries from the Western Pacific regions, just to use this opportunity to discuss the way forward and to establish greater collaboration. So please, those from Australia, Papua New Guinea, we have some people from Japan and also Korea. If you like to participate in this meeting, please, where is Kasim? Yes. So kindly see Kasim after this session and he will direct you to where the meeting will take place.
The meeting is in room D46025.
So is the other building?
Yes, it's now.
After this
so
meeting.
If you can, I'm sorry for the short notice, but if you can, room number D46025.
Okay, thank you. Second, there is a meeting on Tungiasis in W2, room W2. You go up, and there are meeting rooms in the corridor. You see W2, that is for Tungiasis. And then room, SDR PEP for leprosy research projects. This will be in room U2. U2 is also upstairs. And then I think it's a private meeting between Effect Hope and Liverpool also. This meeting will be in D45044. That is in the you have to go out to the other building, D block. So these are the meetings for this evening. If you are not involved in any, I think you can take a break, go for some beer, and that is what I miss. But as the organizer of the meeting, I have to wait till Friday. Yeah, so okay. Tomorrow, I think we have maintained energy and momentum. I know people are getting tired, but try to put some fuel into the car. Tomorrow, we'll be touching on diagnostics and treatment. Please have a nice evening and see you tomorrow.